Students, you do, huh? You are so dark.
Oh no.
Right?
Better.
Okay. Better, yes. So what we're gonna do next? We're gonna introduce ourselves to our students, so then they can know us before start the class. Actually, to start from me, I am associate professor, Dr. Pachani Chutong. So then today I am responsible for being the reader in this topic. Who next? อาจารย์โออาจารย์ชนิดา
Yeah, I think some of you probably know me already, but some of you don't know me and probably never seen me before. So my name is Chania. Yeah, so my major is the immunology. So I'm happy to see you all today.
พระสองแค่น้ำครับ That's it.
That's it.
That's it ประสงค์แค่นั้น
Okay. I think they know me already. I'm Narutai. Okay.
Okay, so me is Vishayaporn Kampom. So I'm from Department of Microbiology and Applied Technology.
Thank you so much for everyone. So student, I sent you the lesson plans in the Google Classroom. Have you seen that for the questions and for the assignments? Everyone
Yes,
see that, right? Please let me know. Everyone see that, right? So then today we will start from the assignment one that I give you 3 questions, and then I give you for the group presentation and group discussion. So hopefully that you guys do assignments and discuss and talk in the class. I mean in the group before come to the class today. We have 3 questions. So then we start from the question one that asks you about how does SARS-CoV-2 virus contribute to immunopathogenesis in COVID-19 patients. That Ajahn Chaniya and Ajahn Naluthai are responsible for this topic. So start from these questions, and you guys have allowed 30 minutes for this question. I mean presentation and then question and answer and class discussion. All right. Group 1, you guys are ready? Yes. You have the PowerPoint and any material that do you have or do you prepare?
Yes.
Yeah, please go ahead. You have 30 minutes.
So who will show the PowerPoint so I can make you as the co-host so they can show the PowerPoint. Group 1.
Let me open the PowerPoint then.
Okay. Natapum.
ครับผม My computer is a bit slow, so just bear with me. Okay. Okay. So that, and then share the slide. Yep. Okay. Okay. Can everyone see the screen?
Sure. Yep.
Okay. So shall we start then? Yep.
Yeah. Go ahead.
Okay.
Good afternoon, Ajahn and my friend. Our group will get a topic in the immunopathogenesis of the SARS-CoV-2 virus. For the SARS-CoV-2 is a single-strand RNA with the envelope virus. It is a member of the beta coronavirus, and when look this virus under fluorescent microscope, the researcher found that this virus is a spherical shape with spine that look like a cow, and this virus composed of the non-structured protein, including the essential enzyme and the structural protein, including the S protein, which are the spike that the virus used to attack the host cell receptor. And the virus has a membrane glycoprotein or M protein, envelope protein or E protein, and the nucleocapsid or N protein.
Yeah. First of all, I think I would like to make sure something and correct in between your presentation.
Okay.
It would be okay. Who was going present? Is Mongkon, right?
ผม
Yes.
Yeah.
So when you say about you using the fluorescent microscope to see the structure of the
sorry
sarcophagus.
ครับ not fluorescent ครับ sorry electron microscope ครับ sorry ครับ
All right. So be careful about what when you say, okay?
sorry ครับ
And one more thing that I said, it is a grad class. So then you present with your, how to say, the knowledge that you are synthesized already. Don't come with us with the content. So then it's no score for you guys. So then you come with the answer that you already have that knowledge. Don't come with the basic, like what is the SARS-CoV-2 blah blah blah, because the question is immunopathogenesis. All right, you guys have 20 minutes for presentation. If you fail this one, that means you fail for this class, and then you need to do it again. It's not come in the content what is no. You have knowledge, you synthesize it, you integrate the knowledge, and then you answer my question. This is how you study PhD or master. Yeah, go ahead.
and because of the virus use the S protein to attack the is used S protein to attack the receptor of the host cell. We will focus on the S protein. The S protein can divide into the two groups. First one is the S1 subunit is bind to the is the is contain the particle that or antigen that can bind to the receptor on the host cell. And the second one is the S2 subunit or viral fusion entry is used for fusion and entry into the host cells. Please go next slide. This slide show the after you infect with the SARS-CoV-2 virus, the virus will they have the S protein on the spike to attack the ACE or angiotensin converting enzyme. Converting enzyme receptor. These are the receptor for attack and entry the virus into the host cell. And they use the TMPSS2 as a protease to cleave the S protein of the SARS-CoV and initiate the process of entry the host cell. These are the physiology of the SARS-CoV infection in the cell. The after the virus bind to the ACE receptor, it will enter into the host cell and it release the RNA into the host cell for replicate and for replication. And the viral RNA are detected and recognized by the PRR and stimulate and then they stimulate the TMEM173 to induce the cytokine production, which are the IL-6 and type 1 interferon, including interferon alpha and beta host. And the PRR also activate the CASP11, which respond to the excited intercellular danger signal, causing the pyroptosis, which involved in the inflammation also. For the toll-like receptor pathway, which are the one of PRR molecule, the toll-like receptor 3 and 7 and 8 play role in stimulate type 1 interferon release via activated the IRF3 and in F kappa B. And the toll-like 4 is recognized the oxidized phospholipid produced by the SARS-CoV-2 infection, which activate the pro-inflammatory cytokine releasing.
Okay, now we are in the part that we will answer this question about the immunopathology of the COVID-19. Actually, I will start with the overview of the immunopathogenesis, and then the other of the members will be clarify you about the mechanisms. So I will start with the immunopathology of the COVID-19. First, the immune pattern of the COVID-19 will be included with the lymphopenia, the lymphocyte activation and dysfunctions, the abnormalities of granulocyte and monocytes, the increased production of the cytokines, and last but not least is the increased antibodies. For the lymphopenia is a key features of the patients with COVID-19, especially in the severe cases. The CD69, CD38, and CD44 are highly expressed on the CD4 positive and CD8 positive T cells of patients. And also the virus-specific T cell from severe cases exhibits a central memory phenotype with high levels of INF gamma, TNF alpha, and also the interleukin 2. However, the lymphocytes show an exhaustion phenotype with programmed cell death protein PD1, T cell immunoglobulin domain and mucin domain 3 or TIM3, and killer cell lectin-like receptor subfamily C, member 1 or NKG2A upregulations. For the neutrophil levels are significantly higher in severe patients, while the percentage of eosinophil basophil monocytes are reduced. Next, for the cytokine level, increased cytokine production is another key characteristic of the severe COVID-19. Most severe COVID-19 cases and exhibits an extreme increase in inflammatory cytokine, including IL-1 beta, IL-2, IL-6, IL-7, IL-8, and IL-10. Also the GCSF, the GMCSF, the IP10, the MCP1, and also another component that will representing a cytokine storm, which will be clarify you about more understanding. Next is the IgG levels. No, sorry. Uh, the last is the IgG levels also increased, and there is a higher titer of the total antibodies. Interestingly, there was a study found that an increase of the IgG response is closely associated with the disease severities, indicating a simple complementary marker to discriminate between severe and non-severe cases. Next slide, please. And the next slide, we're talking about the potential mechanisms of the SARS-CoV-2 induced immunopathologies. First, on the left hand, on the above left hand side is a potential mechanism of depletion and exhaustion of lymphocytes. First is ACE2 receptor expression on lymphocytes, especially on T cells, promote the SARS-CoV-2 entry to the lymphocytes. Two is the concomitant increase in inflammatory cytokine levels, promote the depletion and exhaustion of the T cells. Three, the SARS-CoV-2 directly damage lymphatic organs, including the spleen and lymph nodes, inducing the lymphopenia. And four, the increased lactic acid levels inhibit the proliferation and dysfunctions of the lymphocytes. Next, on the right hand is about the increase of the neutrophils. It actually comes from the lymphopenia that may lead to the infections with microbe, further promoting the activation and recruitment of neutrophil in the blood. Third one is on the below left side about the cytokine storms. The potential mechanisms of the cytokine storm inductions. First is the CD4 positive T cells can be rapidly activated into the T helper 1 cells and secrete the GM-CSF, further inducing the CD14 positive and CD16 positive monocytes with a high level of the interleukin 6. Second is an increase in in CD14 positive interleukin 1 beta and monocyte subpopulation promotes the increase of the interleukin 1 beta productions. And the third is the T helper 17 cell producing the interleukin 17 to further recruit the monocytes, macrophages, and neutrophils, and stimulate other cytokine cascades, such as the interleukin 1 beta and interleukin 6, among others. And for the last immunopathology is about the ADE, or an antibody-dependent enhancement. A neutralizing monoclonal antibody targeting the virus can enhance virus entry to the cell via the Fc region of the antibodies and bound to the Fc receptor on the cells. This correlated with disease progression and poor outcome of the patient with COVID-19.
For the reason that in the COVID-19 patient have increased neutrophil, is come from three reasons that Nashat said. The first is because of in this COVID-19 patient, they have lymphopenia that cause the lower of the lymphocyte. So it lead to the neutrophil and lymphocyte ratio was higher. And the second cause is because of they have infection in the patient that neutrophil respond for the infection. And the third reason is the neutrophil are promoted and activate and recruit, and our body recruit more neutrophil to clear up the infection. So this three is the reason that why in the COVID-19 patient have the increased neutrophil, especially in the severe patient. This part is talking about the macrophage response to the infection. The macrophage respond to the infection by using the inflammatory molecule which eliminate the pathogen, but the dysregulated macrophage can be damaged to the host or our body, especially in this COVID-19 infection. The hyperinflammatory is the major cause that lead to the severity and mortality in the COVID-19 infected patient. So there are many mechanisms that contribute the hyperactivation from the monocyte derived macrophage and hyperinflammation in the COVID-19. First is the delay of production of interferon type 1. When the virus infection into the epithelial cell, if the delay interferon type 1 response, it can lead to the enhanced cytopenic effect and the increasing of sensing of the virus, and this can enhance the releasing of the monocyte. And then the monocyte differentiate into the pro-inflammatory macrophage to the JAK, which is a JAK signal and activate the STAT pathway. And not only this, the activate NK cell and T cell also promote the releasing of the monocyte derived macrophage to the GM-CSF, tumor necrotic factor, and interferon gamma too. And another mechanism is related to the oxidative state, which the oxidized phospholipid accumulate into the lung infection through the toll-like receptor for TRAF6 and NF-kappa B pathway. And the virus sensing can trigger by the toll-like receptor 7 activated to the single-strand RNA. And for this, we can suspect that interferon type 1 induce the expression of the severe acute respiratory syndrome of the COVID receptors, and the virus can access to the macrophage by using the ACE2 or the CD147 attraction, and it activates the NLRP3 inflammasome and can lead to the secretion of the interleukin 1 beta and interleukin 118, and the interleukin 1 beta can activate the toll-like receptor 4 also, and the engagement of the Fc gamma receptor by the anti-spike protein LGG immune complex can increase the inflammatory activation, and these all mechanisms can active the monocyte derived macrophage, lead to the cytokine storm releasing by the amount of pro-inflammatory cytokine. So next.
Yes, for this slide, Nashat said in the previous slide, so go to the next slide for further finding. Next slide. Yes, for further finding SARS-CoV-2, I will present in the model of cytokine storm. SARS-CoV-2 interact with ACE2 receptor and get into the monocytes, and it begin replication itself and create more virus. And monocyte release interferon type 1, which are interferon alpha and beta. Type 1 interferon are very great at actually control nearby cellular response. So they tell nearby cell to increase expression of MHC class 1. And for another part is immature dendritic cell pick up the virus and take it to lymph node because it will be gonna be mature and present virus antigen to CD8 to become activated CD8 and kill virus in the site of infection. When the cell die and nearby monocytes and macrophages will pick up the dead cell and become activate themselves, produce cytokine and also pick virus and become infect. The macrophage produce cytokines such as interleukin 12, TNF, interleukin 1 beta, and create leaky endothelium virus and create leaky endothelium cells so virus can get out and get more this cytokine. Yes. For this slide, I will show you that an interleukin 6 is the key role to cytokine storm. Interleukin 6 has many of physical function including production of acute phase reactant, evaluate hyperinflammatory, and all of these cytokines, interleukin 6, interleukin 1 beta, interleukin 12, TNF alpha, can stimulate the virus and occur inflammatory cycle again and again, and all of this is gonna be go to cytokine storm. Yes. In the next slide. For this slide, SARS-CoV-2 infection of monocytes, macrophage, and dendritic cells result in the activation and secretion of interleukin 6 and other cytokines. Interleukin 6 can signal through two main pathways referred to classical signaling and trans signaling. The first is in cis pathway. Interleukin 6 bind to membrane bound interleukin 6 receptor in the left side of the presentation and complex with GP130. The GP130 is expressed whereas membrane bound interleukin 6 receptor expression is restricted to immune cells. Activation of 6 signaling result in lymphocyte change, which can contribute to cytokine releasing syndrome. For in the next signaling is trans. High circulating concentration of interleukin 6 bind to soluble form of interleukin 6 receptor forming a complex with GP130 and to be mediated by JAK and STAT3. So for this signaling is activating in cell and do not express in another part, that is this signaling that not expressed, such as epithelial cells. Yes, this result in systemic cytokine syndrome involves secrete of vascular endothelial growth factor. Sorry, we go to the next. Yes. for the trans signaling is leading to VEGF increase, MCP1 increase, interleukin-8, interleukin-6 more increase. So it's going to be cytokine storm. Yes.
Okay, for the last part, I will talk about a bit about the exhaustion of the lymphocyte. So I will skip since there are some people say about explanation about briefly about this. So I will focus on the active immune response. So basically, as we could see, SARS-CoV is like every infection in as viral infection. So it will trigger cytokine production. Cytokine production from the T helper cells, Treg to regulate the inflammation. Cytotoxic T cell for killing the infected cells, and also for the increase the antibody production, which includes the B cells as well. And then as you could see that like it's normal. I think it's the same response with any other virus actually. But what happened is though, actually in most people, not most, but some people who got very severe COVID infections, what happened is the lymphocytes got decreased, which is one of the reasons why we need to talk about this cytokine storm first, because cytokine storm is one of the pathway that leads to the CD4 or CD8 cell exhaustion. Like in this slide, I got a review from Nature. Actually, I forgot to put the reference in. But actually, in normal, in my disease, what happens is there is the secretion of IL-2 and type 1, type 3 interferon that leads to the T helper cell proliferation, and that leads to the immune response in terms of the cytotoxics and in terms of the building antibodies. But what happened with the severe disease, as we know that the secretion mostly is IL-6 and IL-10, and the IL-6 has a role in increased stimulation of T helper 1 cells, but it's not increase the proliferation though, it's hyperactivated. So what happens is you still get the immune response, but with more pathologic effect as well, which I still need to discover more what happened is. But anyways, steps like that. One of the interesting cells though actually is CCR6 Th17 cells. I think as other of my group mates tells that there is the differentiation of Th17 increase in terms of differentiation. So what happened is though in I think in coronavirus infection there is that like signature that you will get the expression of CCR6 with that expressed in Th17 cells. Th17 cell, even though it has a role in clearing pathogens, but I found that like there are some papers that it's a pathology that causing inflammation and autoimmune system autoimmune disease as well. So that's kind of keep in mind what might happen. For the CD8 T cell exhaustion is a bit easier than CD4. So basically, again, IL-2 type 1, type 3 activates the T cell, leads to clonal expansion, which leads to the termination of the viral infection. However, for the severe disease, though, so the IL-6 instead that stimulate. But what happened is when CD6 stimulated, CD6 also stimulate the cell death pathway as well, which might be the cause of reducing of the CD8 T cell pool as well. So that's why it's the CD8 drops significantly, and that leads to the pathology that people got more infection because of the cytotoxic T cell couldn't be produced, and that leads to more inflammation, more infection of the viruses. So one of the signaling pathway that I investigate is the IL-6 stimulate the AKG2A expression in CD8 and also in NK cells as well. So basically, CD8 and NK cells are involved mainly for anti-COVID-19 response. So basically, COVID-19 infection increased the production of IL-10, IL-6, and that leads to increased neutrophil, and that because it induces more inflammation. But most importantly, though, when we look at the not an adaptive response, is increase the expression of NKG2A, and that is the inhibitory signal to inhibit the immune response. So what happened is it's reduced the effects of the NK cells and CD8 as well, and that also leads to the immune response to be lower than normal patients. Oops. The last one is the antibody-dependent enhancement. So I searched a bit about the disease actually. So the ADE of disease is the condition where antibody produced by B cells and that cause progression of disease instead of protecting the infection or protecting the cells. Most of the time though, when we talk about the monoclonal antibodies, it attacks the pathogen and that blocks the let's say the blocks the infusion for endocytosis of the viruses, or in terms of the bacterial, it not optimize, maybe like flag that pathogen and leads to the immune response. But what happened is actually in terms of the disease progression, so the antibodies might be impaired. So even though it's bind to the viruses, the protein itself, there is a viral protein fusion as well. That's because of the virus has like lots of mutations, drift mutation, maybe that leads to the mutation as well. It also the fusion of the cell of the viruses as well by coagulating all the virus together, and that leads to that the antibodies couldn't be blocked by the ACE receptor. So basically, the antibody can't block the complex between the receptor and the viruses. But more importantly, what happened is the antibody binds and that enhance the viral fusion instead. I think one of the slides showed that it fusion with FC receptor on the cells. But in this paper, they said that they just enhance the infusion, but they didn't say the pathway what it is. So I think it might be FC receptor as well. So in terms of clinical application, so this is a summary of everything. Actually, I might be a bit fast, so sorry about that. So it affects the lymphopenia on the infection. So basically, the adaptive response got mute, so it got depressed. So that happens to be T cell, NK cell reduction, especially the CD8 and NK cell that leads to more infections due to there's no soldiers to defend the bacterial infections. The second one is I think it's the most important one is a cytokine storm actually, because cytokine storm enhance the inflammation response, and that leads to the organ dysfunction due to I think vasodilation as well. That leads to overperfusion, and that leads to the organ failure as well. So mostly, as you could see, there is the respiratory failure. There could be some dysfunction with the liver, so liver needs to work faster. So the enzyme that relates to the liver increase as well. And then the heart needs to work faster as well, since there's lots of vasodilation, so it needs to be working more. So the marker CK and LDH is increased as well. So I think that's about it. I think. Yep. So if there are any questions, just ask. So I will be happy. Whoops, sorry about that. I'll be happy to answer any question. Thank you.
Thank you, our students. So it takes around 45 minutes. That is, it's too long for the presentation, but it's okay. Yeah. So then maybe question from student and your friend first before staff ask in the content of the immunopathogenesis. Any question from the student? You listen your friends and then you can answer yourself that what is the immunopathogenesis in COVID-19. If not, please ask the question because it's a concept. Any question from student? No?
I want to ask to clarify more about the T cell exhaustion. What
Yep.
is about?
Yep.
I think this slide. Yeah.
I think go to the one that Nashat presentation. That one is a good one. Yeah.
This one.
Which one?
I think this one. This one.
The
Yeah.
slide that you have. No, the slide that you summarize all. Yeah. No.
Yeah, this
Yeah,
one.
this one. Yep.
Yeah.
The question is.
The T cell exhaustion. What is about that?
So when you say about exhaustion, it means that the number of T cell decrease or the efficiency of the cytotoxic effects of the T cell got reduced. I think that's what in terms of exhaustion. So it exhausts by the cytokine storm, and that leads to immunopathology in terms of the lymphopenia, because they cannot they're weak soldier basically, so they can't do anything.
Yeah, but the exhaustion is come from only cytokine storm. Do they mention any mechanism that support only the cytokine storm that induce T cell exhaustion from your reviews?
For now it would be that, like, I got some evidence that the T cell got directly infected by the COVID virus as well, but I still haven't find out the paper specifically how it works. So, but yeah, most of the T cells had ACE2 receptor as well. So instead of they fighting for it also is a target for the COVID viruses as well. So I think that would be one of the mechanisms that instead of cytokine storm, it also they got infected itself with the viruses, and that leads to the lymphopenia.
You mean T cell has the receptor for the COVID-19 to bind?
Yeah. So it had, I think it had like nearly 90% expression. I still pending on the research actually, because there's so many research about the ACE2 receptor on the tissue, but not many on the immune cell itself. I think that's the problem that I had so far. But I think it expressed the ACE2 receptor. Yeah, definitely.
Thank you.
Do you think for the cytokine storm that could induce like down regulation or up regulation of ACE2? Do you find this evidence from your reviews? The cytokine storm could induce up regulation or down regulation of ACE2 receptor. Anyone find this evidence?
I found it the opposite way actually.
Yeah.
It's the ACE2 got down regulate by IL-6 production, but that's not like, but that as the cells got produced the IL-6, and that leads to ACE2 down regulate, not down expression, the expression, but I still try to find whether if the IL-6 got stimulate to the cells and it's induced ACE2 receptor. But yeah, still questioning. But any my team member could help.
Yeah.
I think the student need to clarify the term, because I don't think the T cell exhaustion is it's not, it doesn't mean that the number is decreased, but its function is, the T cell cannot function as normal. That's the meaning. So the mechanism need to be, it need to be clarified, because now you just talk about lymphopenia, right? But actually the question is about the exhaustion.
Yeah, I was about to say something to, with Ajahn O, because exhaustion means tired. Yeah, and it could be the number are the same, but the function is reduced, right? So, and it could be either direct infection from the SARS-CoV-2 or either cytokine storm. Cytokine storm, what do you think? Why T cell get exhaustion when they have cytokine storm? Why?
I think it's because the T cells infected and is released more cytokine is stimulate around and around and can be T cells exhausted. I think like that.
You mean
Is
that
that
T cell work too much and then get exhausted?
no? She mean T cell get infection, right?
Yes.
No, one thing is T cell get infection. Another thing is cytokine storm. So when we have cytokine storm, the patient has cytokine storm. How T cell get exhausted?
Maybe they
Maybe
they have focused on the proliferation only, but not the function.
They produce too much cytokine and then they get exhausted. That would be one mechanism that I can think. Because the cytokine storm itself is not just only come from the innate cells, it's also come from T cell as well, cytokine that release too much in the patient, right?
But what I think is actually on the slide you can see the markers there, right? There are a number of markers, and those are the thing that you have to relate to a T cell exhaustion. Because yeah, especially the one on the left, you can see the PD1, right? So this thing you have to explain in the mechanism, but you didn't mention about it. You mentioned about NKG2A. Yeah, but PD1 is one of the important markers that involved in the exhaustion of T cell.
Yeah, that's why I would like to ask that. Where once cytokine storm happen in the patients and then T cell exhaustion happen in the patient. So then how cytokine storm do something with the T cells. So Ajahn O help for PD1 and NKG2A or TIM3. You see upregulation. So then how can you explain the upregulation of PD1 and KG2A for the T cell exhaustion? If you know the function, then you can think more. Even the article don't mention that you can think and explain us why T cell get exhaustion or tired, as Ajahn Shriya mentioned, for PD1 upregulation or TIM3.
Okay, for the PD1, actually the PD1 is the protein on the surface of the cell that regulate the immune systems, which it is used to down-regulate the immune system and also the promoting of the cell tolerance by suppressing the T cells inflammatory activity to invent the autoimmune disease. So I think if after they have over upregulation of the PD1 protein, it could be just like over suppressing the T cell and making it T cell exhaustion.
You almost there when you think about the immunopathogenesis and immunoregulation. It's the concept of undergrad student. So from your presentation, you just talk about how virus trigger the host, blah, 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 and cytokine storm. All your presentation, but you never mention about immunoregulation. Do the patient that get COVID-19 have the immunoregulation to regulate, to do something with the cytokine storm that the virus triggering? And that's why PD1 is upregulation. Can you explain this? Can you draw this picture? You're almost there. Why in our body, if we get a SARS-CoV-2 infection, why T cell upregulate PD1 for what? You think?
For proliferate and release cytokine
Are you
and kill
sure?
I think like that.
Now, Nashat, can you try again? You
Yeah, Nashat almost there, but the keyword is immunoregulation. You're almost there.
I will try, but I'm not sure it's quite correct.
Yeah.
Or
Yeah.
it's from my understanding, because the cytokine storm can lead to many pathogenesis, right? So the body have to regulate or to stop the inflammatory activity then. So they have to express more some protein that can reduce or inhibit the inflammatory activity. So they upregulate the PD-1 protein to stop to cope with
Stop
the
what?
cytokine.
PD-1 on what cell to stop what cell? Then you explain that.
I think that PD-1 is not upregulated for inhibit the activation of the T cell.
How? PD-1 bind to what? You come like a fragment. Try again.
Because maybe the PD-1 is on the regulatory T cell, right? So
Ajahn Oh, he's smiling now.
um,
Go ahead.
PD-1 is not only in the regulatory T cell, so it would be in the sense of T cell exhaustion. It means that that cell cannot work well. So PD-1 somehow is also expressed on any other T cells. Yeah, both PD-1 and TIM3. Yeah. So if you think in the point of regulation, these T cell that express PD-1 and TIM3 can also inhibit or stop the macrophage or dendritic cells or antigen presenting cell to activate themselves, right? Yeah, so it's almost there, but just you need to link the information.
Yep.
Yeah, and actually the keyword for this is the immune checkpoint, right? Because if you have PD-1 and PD-L1, the interaction of this will induce tolerance. That means the cell cannot respond to the activation, right? So yeah, this molecule is kind of very important in the precision medicine.
Yeah. So I think to get the answer for the T cell or lymphocyte exhaustion, one mechanism is here is the upregulation of the how to say, it's not inhibitory receptor, but it's kind of receptor involved with the immunoregulations, and that's effect for the function of the T cells that induce T cell exhaustion. I think that is the conclusion that we can summarize for the knowledge so far that we have from the article. Yeah. What about lymphopenia? Always NK cell is reduced in the patient. All lymphocytes from the picture in this one, CD8, CD4, B cell, NK, they are reduced in the severe case.
Yes, but I have found some study. They only remarkably reduce of the the lymphocyte is the CD4 positive T cell, the CD8 positive T cell, and NK cell. But for the B cell is not. They have found that the number of the B cell is still in normal, but the function of producing the antibody that's specific to the virus is downregulated, but not for the amount.
More question from students to make sure that you guys are with your friends.
So please clarify more about the increasing of the neutrophil.
High number of neutrophils.
High number, yeah.
Yeah.
High number of what?
Neutrophil.
Neutrophil.
Yeah, our population it seems like reduced, but neutrophil is increased. So what happened?
I'm not sure that answer that I guess correct or not, but from my understanding that in this increasing of the neutrophil is caused by the they they have many inflammatory in the our body, and neutrophil is responsible for the infection too. And I think the mainly cause of the neutrophil increasing is from the like it's increased because of the ratio of the neutrophil and the leukocyte ratio is like if you have the low number of the lymphocyte, and it can lead to the neutrophil to the lymphocyte ratio is increasing. So yeah, I think that in this case is the high neutrophil because of this NLR ratio, NLR.
You actually present one slide and it show quite clear. So the proportion of or the ratio of the cells is something, but we talk about the number. When we talk about the number means that you have the reference range, which is normal, right? But
Yeah.
how neutrophil is increased up, and you have one slide to explain that why
Yeah.
neutrophil is increased. It's not all the case, but just some case that it might increase, right?
Yeah.
Okay, explain
I
on that.
would like to wrap up. No. Yes. Actually, for the neutrophil levels, it is significantly higher due to PBS. Can you go to the slide that?
This one.
Yeah, right. Yep.
Yeah, because as you see, in the patient with the COVID-19, they will have a lymphopenia, right? So it's meaning that another immune cell may be downregulate and cannot function well. So the microbial, which is the normal flora or something, can be come from inside or the outside, can be just like more easy to infect the patient, right? So after that, if you got that infections, the first response, the immune response is to upregulate the neutrophil to cope with that infection or to kill that patient, the kill that infection. So why the number of neutrophil is high.
May I ask? I'm just curious. Actually, even me, I don't have the answer, but many students can help.
Okay.
Do you think it's possible that neutrophil is increased in the patient? It come from cytokines because come from inflammatory cytokines. It's possible. If yes,
Yes.
could you
Yes.
explain? Yes, yes, yes. Okay.
Yes, it is.
Who think is yes? Can you share with me? I actually I don't have the evidence, but the idea just come up. But if you have the evidence, can you share? Yeah.
Yes.
Slide in this presentation, it is, can you show cytokine, the one previously we were explaining, the slide which shows cytokines
Which one? This one.
again, back again.
Back.
The one again, the one lymphocyte we are describing. Yes, not this one, the other one. Yes, in this one.
This one.
No, back. Among
Which
the,
one?
no, that again, you came to the same.
This one.
Yes, this one.
This one. Okay.
And among the cytokines, here is the growth factors. Growth factor for granulocyte colony stimulating factor, granulocyte monocyte colony stimulating factor. And these are the factors which led to the proliferation of more neutrophils.
So you think that because come from the cytokines, some cytokines that produce in the
Yes.
part of cytokine storm in the innate immunity that
Yeah.
produce something like core neutrophils come up. That's why it show high level or increase. Yeah, I agree with you. But your friends, you have more answer? Just share the idea, because this is new. But share with the knowledge. What else that could induce high neutrophils?
I think so with Chimera because when cytokine storm is occur, it maybe induce more neutrophil to release the cytokine. Oh, sorry. I think like Chimera.
Yeah.
When you look at the cytokine, you can see that the GCSF and GM-CSF actually act on bone marrow to help produce the new myeloid cell, not only neutrophils, myeloid cell, including macrophage and dendritic cells. And then when you look at the IL-8 or IL-17, this is the cytokine that help recruitment of the neutrophil. So you produce more and recruitment more so that neutrophil might increase in the blood circulation.
Yeah. Everyone seems like agree with this, right? All right. Any question before we move to the second group because it's running out of time? But any question or discussion? Yeah.
I want to ask. I want to know the source of cytokine initially because when they are giving presentation, I was little confused.
About cytokine?
During the initial infection, what is the source of cytokine? Did you hear me properly?
You mean when
I
they
think
call?
What is the source of cytokine during initial infection? And I want to know cytokine. I mean, during initial infection, cytokine involvement is which type of immune response?
Okay.
You have the slide actually, the one
Yeah.
that talk about monocyte.
Monocyte.
Start from the innate and then to explain to adaptive one.
yes, I will try. Okay. And SARS-CoV-2 interact with ACE receptor and get into monocytes. Shivla, do you want to mechanism of this like?
You go to the one that you show the
Not
monocyte.
mechanism. I just want to know what is the source of cytokine, initial source of cytokine.
Right.
From where
Go to
cytokine
slide
come
of
from?
Starting from infect the epithelium cell first.
Yeah, this one. You start from infection, what happened, in the what, and then cytokine is feedback to what, and then in the what, that is the state of the infection. Start from the virus come to the epithelial cell.
It's infect into the epithelial cell and the interferon type 1 activated and that interferon type 1 lead to enhance the releasing of the monocyte and then the monocyte will
Wait, stop. You have the viral infections. You mean that virus induce what cell to produce type 1 interferon? If you think it's type 1 interferon, come for step 2 step to make sure that your friend can follow. You have virus infect to the cell. What cell produce cytokines and induce forward to the next step? Go to the step and mechanism. Look at the
virus get into the monocytes.
Yeah.
And monocytes. Or
Start
virus
from epithelial cell, that's the virus, right? Alveolar epithelial cell.
Yes.
You understand this? Yeah.
Start from the epithelial cells.
Right, because virus it infect the cell first, right?
Yes, virus interact with ACE receptors
Mm.
and get into the monocytes. And monocytes.
How to get into monocyte? Natapum, could
Yes, yes.
you have
I mean,
you have the immune response, right? I think you can help your friends. Virus infect and then produce what, before you jump to the monocyte.
okay. So
Mm.
I will try my best.
Mm.
Actually, so basically, so when the epithelium cell got infected by the virus, what happened is the PRR try to recognize it. So basically, the toll-like receptor, the host defense mechanism, like this signaling leads to the production of the cytokine,
Right.
and the
Exactly.
cytokine released from the epithelium cells, and that signaling to the monocyte, or actually macrophage as well, in within the ovaries. But in terms of the monocyte, it so basically it the cytokine release from the epithelium cells and then reach to I think it needs to be chemokine first. So basically, it's vasodilation. So it recruits the monocytes or the neutrophils into the infected site, and that leads to the initiation of the reaction. So basically, the type 1 interferon response. So type 1, so the interferon binds to receptor, and that leads to signaling, and that's also the others chemical as well. I think I think that's answer the source of the infection, and that's I think it's primary for the cytokine production is start with the epithelium cell first. I think.
Yeah,
Are you sure? Are you sure epithelial cells will produce cytokines? Because when I was doing my studies, I came to know the only cytokine epithelial cell will produce, that is tumor necrosis factor. That is also because of the association of virus S spike with the enzyme which is related to tumor necrosis factor. In this presentation, you people have the same slide which I have used in my presentation, my one. And in that slide, it clearly says that once interleukin get activated by transist pathway, it will allow epithelial cells to release the epithelial cells which don't have receptors also to induce cytokine, to respond to cytokine. Then how will it is possible to epithelial cell to release cytokine?
So,
Initially,
maybe I can help a bit because you think about the
I asked this question because initially when the second person was presenting this, he said cytokine release is from CD4 cells. Initially, there is no CD4 cells. This is the innate immune response. Am I right?
yes.
And CD4, so that's why I am asking. And from same slide, and while presenting this, clearly mentioned that the interleukin 6 have a pleiotropic effect, and it will induce epithelial cells also to release it.
Yes. So, yes, you're right. But just to add up Natapum and also Chimila, because once the virus get into the lungs or in the airway, so of course the first barrier they will face to these epithelium cells, and then get enter into the epithelium cells. Also that in the lungs you have another immune cells as well, especially alveolar macrophage and also dendritic cells. These alveolar macrophage and dendritic cells, they can also produce pro-inflammatory cytokine such as TNF-alpha, as you mentioned. right? IL-1 and IL-6. And then that was the initial. The more initial is the epithelium cell produce interferon alpha. This interferon alpha also can work and help monocyte macrophage or dendritic cells to fight against the virus as well. Yeah, the initial cytokine, of course, come from innate. Yeah. Okay, you correct. And also Natapum and who else? Nachat try to explain, but it's not just the point that you want. Yeah. Okay.
But to add up in that, for toll-like receptor, Natapum mentioned epithelial cells have toll-like receptor for this, but I'm not sure which one. But you guys present like a TOL2, TOL3, right?
I think it's
That
Toll
express,
3. Yeah.
yeah,
OT and show 7/8.
that express on epithelial cell also, right?
In from my understanding in the picture is that the receptor. I'm not sure. Maybe in the cell, don't not on the surface. ครับ how can I say?
Toll-like receptor 3, I searched it actually. It's actually expressed in the epithelium cells in the airway as well. So that's why Mongkol tried to say about the toll-like receptor 3, I think. And I think it's one of the pathway that kind of directly affects with the viruses infection, I think.
Yeah, and that the outcome is induced inflammatory cytokine also that Ajahn Chaniya mentioned. So that maybe is the answer your friend question is initial cytokine is come from the innate immune response. Yeah. More question before we go to the assignment question 2.
I have some questions.
Yes.
Yeah.
Could you go back to the slide that shows about the T-cell exhaust?
Which one? This one. This one actually.
No. The slide that the first of the first question discussed. That shows the go back, please.
This one or
No.
one?
Yes, this slide.
Okay. There's a delay going on.
Okay. I'm curious that why monocytes is decreased because in the presentations I have seen that you say that monocytes is or the macrophage is activated or the increase in cytokines also activate the macrophage while the monocyte is still decreased or why only
yeah, we got the question, but then who gonna answer this question before our staff can help? Yeah, monocyte, why is so like the result like this? Yeah. Who gonna explain it? Activate monocyte. It seems like monocyte function, but then you go to the blood circulation and show monocytes.
Can I try?
Sure.
From my finding is after the infection they got the inflammation, right, and they release the many cytokine and chemokine, and for the chemokine they will call the bring the monocyte and lymphocyte from the blood vessel come out to the infected site or inflammatory site, and I think it's just the reason that why the monocyte is decreased, because we count the number of monocyte in the blood, but and now it's just go out.
Yeah, you mean monocyte get activation and then monocyte move to the peripheral tissue or in the tissue. That why it show low number in the circulation, right? Yep.
Yes.
Yeah, I agree with you. Ajahn Chaniya, you would like to add up in this point?
Yeah, possible.
Yeah. More questions? Because we are, right now it's more than 1 hour. อาจารย์โอประสงค์นฤทัยอาจารย์วิชยาพร you would like to give the comment or share the knowledge for this question before we move to the next question?
Yes, I have one thing. Oh, sorry. Ajahn Ruthai, right?
No, Ajahn Chania.
Okay,
Maybe group 1 can summarize again. Yeah, what is correlated to the patient symptom? Yeah, before we move to the next question, because we discussed a lot detail in the mechanism, in the molecular mechanism, but then you might forgot that what is related to the clinical symptom. Yeah.
Yeah.
So, Ajahn O, what you were about to say?
So actually, what I want to say is the main or the key feature that we see in the patients. For example, the ARDS, we haven't, I mean, it's not quite clear about this. Okay, so yeah, I think yeah, we go to the detail for many things, but the main or the key feature we haven't mentioned about it much.
Yeah, and I want to know too, because right now, right, from your presentation, like the patient have the symptom only the respiratory tract. So then in the severe case, do they have more complications?
Yeah, you present already, but just before we end this, exercise again.
I think it's in the last slide, I think. Yeah.
Okay, you have 1 minute.
So I just summarize. So basically, when the immune cell, especially the adaptive cells, leads to the more infection chance. So you will get like more pulmonary, like that pulmonary disease or maybe infection disease, and that might lead to septic shock, some kind like that. That's one of the severe case. And also when you elevate the cytokine, that leads to the organ dysfunction due to the pathology that increase the vasodilation. So the blood supply cannot work, I think, cannot go to the organ, and that leads to the respiratory failure because of the, especially in the lung, actually, infection and inflammation from infected cells, and the rest are the heart, kidney as well. I think cardiovascular disease is one of the link that also linked to the COVID as well. I think that some patients do have the complication with COVID as well. Yeah.
Excellent.
Yep. So we move to the next questions. Then you guys will have more understanding why some patients have the abnormality of the autoimmune disease or autoinflammatory syndrome. Yep. Who gonna present?
Did everyone see the screen?
Yep.
So in our group, I will start to brief a little bit about systemic lupus erythematosus. Is one of the model of the autoimmune disease. So SLE or the systemic lupus erythematosus is one of the systemic autoimmune that's characterized in a lost immune tolerance and produce autoantibody. The factors may be genetic, environment, stimuli such as infectious or the UV export, and it's causing the apoptosis in the body. If in the bodies cannot clear the apoptosis debris. This device will be appear as the autoantigens. Then they can start the innate immune response following the activations of adaptive immune response. Once that T cell and B cell were represented autoantigens, if they cannot recognize or lost tolerance, it will lead to produce an of the antibody specific to the cell antigens. This autoantibody could by will by both of apoptosis disease and cell molecular in outer parts and occur the immune response lead to inflammation. In worst case, it can develop into the clinical disease, chronic inflammations, and organ failures. From this brief, and as the group one mentions, they have words that say they have one word that same in both of autoimmune and COVID infection is inflammation. So from these keywords, our group will show SARS-CoV-2 infection associated with the appearance of autoimmune and autoinflammation disease. So start with epigenetic dysregulation in SLE. Nunit. Hello.
Next slide.
Oh, okay.
Okay. From my searching, I found this topic. I'm not sure is the articles or review is quite like a mini review and like a they took the other experiment and then the analyze again in the topic epigenetic dysregulations of ACE2 and interferon regulated gene might suggest increased COVID-19 susceptibility and severity in lupus patients. The epigenetic dysregulation in this topic are involved in the DNA methylation. The DNA methylation is a biological process by which methyl group are added to the DNA molecule. And they can change the activity of DNA without changing the sequence. The DNA methylation is one process that controls the expression. One DNA methylation is occurred that DNA become the silence. And DNA methylation involved in many processes such as cancer, aging, and in SLE patients, they found the hypomethylation is widespread, such as DNA methylation signature in interferon regulated genes. And in here, we focus on the X chromosome inactivation processing. Hypomethylation inactive X chromosome lead to abnormal increase in expression level of affect gene. And in the X chromosome, they have the CD154 or CD40 ligand gene. They are a member of the TNF superfamily molecule, and they encode protein on the surface of the T cell that regulate the B cell functions by engulfing CD40 on the B cell surface. They can bind to the CD40 back. They can bind to the CD40 and they can signaling in the B cell activations and F kappa B, which is the pro-inflammatory system. Next slide. Okay, and in the SARS-CoV-2, the ACE2 gene is also located in the X chromosome also. And in picture on the left side, you see there are like two mechanisms at the group 1 already said as the endosome by ACE2 receptor and plasma membrane. So in this paper, they took the information from the previous article in that investigate about the DNA methylations in the lupus patients. In this, they collect the CD4 T cells of the lupus patients, and then they analyze the methylations between the patient with healthy and the lupus patients to see the methylations. And in the left side, show that in the patient with lupus, they are hypomethylation. And then they investigate the ACE2 mRNA expressions, compare between the patient that compare the group, healthy group and the lupus patients. Also found that the ACE2 mRNA expression level are higher than the control group. Next slide. So in here, they conclude that the patients who have the SLE that occur the reduced DNA methylations that cause the higher expression, the AEC2 expressions. And once these patients they got COVID infections, this COVID they also induce the oxidative stress, cause the SLE lupus flare, and then this DNA methylations there also the increase the NF-kappa B and cytokine gene also that also cause the cytokine storm. It means that the SLE patients that got COVID infections they can like more severe than people that got only COVID infections.
So to summarize, to summarize, the first one that you need presentation is the possible mechanism is because from the hypomaturation. So that's why in the lupus patients will have more severity than COVID patient, right?
Yes.
Yeah. Move to the second possible mechanism.
Yes, the second possible mechanism is the loss of immune regulation.
From this picture, this is a good one. So in case of COVID-19, how's the picture look like? This is an autoimmune disease. It look like this. Treg, T effector cells. What about COVID-19? The picture is look like what? The same? Your friends can discuss. Do you think it look like this? In COVID-19, the first group presentation. High Treg, low TF.
I think, in my opinion, it could happen both of the pictures in case of the. It depends on the state.
Severe, let's case severe COVID-19 that your friend presentation, high cytokine storm, blah blah blah. We talk a lot. How the picture look like?
Maybe, in my opinion, I think it's become maybe in the upper, because as we have known that in the immunopathology of the COVID-19 can cause the T cell number is lower, and also the T cell exhaustion. So maybe there is low amount of the T effector cells.
You mean
I
like
think
autoimmunity, right, Nashat?
No,
You
no.
mean balance? But first balance or imbalance, and then if imbalance, it look like what?
Maybe it's not quite balanced, but it's not as much as autoimmunity, because in COVID-19, due to the T cell exhaustion, maybe it quite is, how can I say that, because there is not much T effectors than as same as the autoimmunity, so maybe it's become higher.
But T cell exhaustion, that the T cell that get tired, that is a effector T cell, right?
But the amount,
It's
right?
just no function. But this the picture that they show how it look like
about the
in the
amount, right?
COVID-19. Yeah, which one? High TF, low T reg. Which one do you think?
low T-reg, I think.
No. I want to get answer from Nashat first to make it clear. How it look like? It's having fun. Nobody know, but then you tell me how it look like in COVID-19 from the first group presentation. How it look like?
I think the amount of the T effector cell will be, it will be converted from the autoimmune with the low T effectors and high of the Tregs.
Okay, so look like autoimmunity. Your answer, right?
Yeah.
High Treg. Yeah, but eventually nobody talk about Treg much in the pathogenesis in COVID-19. I want to know from other students. Do you think in COVID-19 how it look like? TF is mean immunoprotection. Treg is mean immunoregulation. How it look like?
Can I try?
Sure.
I think T-reg is lower than the T effector, because T effector have to eliminate the
Virus.
COVID-19, yeah, and then recruit to the site of damage.
So imbalance,
Yeah.
high protection,
Yes.
and then induce to cytokine storm, but low immunoregulation, right?
Yeah, right.
Okay. More answer from student?
I think for me, I think like Canton, but not only the T effector that affect to the virus, but they also high number of the T effector that affect to the other cells how can I say, for Treg that affect to the auto uh, on the cell of patient with cause from the autoimmune also.
Okay, but the answer is high immunoprotection from TF, low immunoregulation from Treg. All right. Yeah. So please remember your answer. Then your friend gonna present and finish, and then I will ask again. All right. Yeah. Go ahead for group 2.
Okay, the next mechanism that can let SARS-CoV to induce autoimmune disease is the molecular mimicry. First of all, I will give the descriptions of the molecular mimicry first. Is the molecular mimicry describes as the structures similarly between foreign antigens and the cell's molecular. Once that foreign antigens presents to induce a production of antibody, this antibody can cross binding to a cell molecular, so it's mainly to contribute immune response to cell. And they have a report that molecular of the SARS-CoV-2, some molecular of the SARS-CoV-2 is similar to the human protein. For example, in this report, SARS-CoV-2 protein ORF7b is similar to the human protein OR7d4, which locate on the olfactory receptor, and a nucleocapsid phosphoprotein from the SARS-CoV-2 is similar to SLC12A6 in the human protein, which locate on the endothelial cell like heart, brain, kidney, liver, and lung. Moreover, they show another report that they have three molecular from the SARS-CoV-2 that mimic to the respiratory pigmaker neuron. What is the respiratory pigmaker neuron? Is a neuron with the intrinsic ability to generate rhythmic bursts that emerge through the voltage, time, and calcium-dependent ion path. So that means if they present the molecular mimicry of the SARS-CoV-2 and induce to produce antibody, if this antibody bind to the neurons in our body, maybe they can affect to the control of the respiratory, and resulting in the patients can and resulting in the patient increased risk of the hypoxia. By the way, the molecular mimicry in SARS-CoV-2, it's just a hypothesis. They didn't already release the fully article that prove the mechanism yet. And Ajahn have any comment in this topic? Or we go to the next.
I don't have, but Ajahn Chiniya, Ajahn O, and Ajahn Ni about molecular mimicry.
Sorry, can I ask for some result from the next, then the previous slide? Can you go back? Yes, this one. So you said that virus have some peptide similar to the human peptide, right? That localize into uh, various tissues. So could you give me the reason, the result? How if the autoantibody produced, for example, for protein, the first one, and the target is the olfactory receptor, what is the symptom that we will see in the COVID-19 patient?
Actually, the ones of the clinical manifestation that can found in the SARS-CoV-2, that's the patients lost the sense of the smelling. So I think if 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 in the if the molecular mimicries they occur the autoantibody which are specific to OR7d4 in the human protein which locate on the olfactory receptor. it could be destroy this receptor and lead the patient loss the ability to smell.
Yes. So you might heard that some patients have the symptom like they can't smell anymore. Like, and for another one, the endothelial cell. Do you think what it leads to the symptom that might cause the systemic inflammation or tissue damage?
Yes, if they appear the molecular mimicry that are specific to the human protein on endothelial cell, I think they could be induce the inflammations in every part that they have endothelial cell, and maybe the resulting is maybe the upregulate of the coagulations uh, pathway too, and it might from the it might form thrombus or thrombins. If these thrombus they have like the appear in the narrows of capillary, maybe or the veins, maybe they appear signs of deep vein thrombosis or DIC.
Yes. Okay. So it's also, as you said, that endothelial cell is the cell that lining on the vascular, right?
Yes.
So anywhere, any organ that has vascular, if the autoantibody attack on the endothelial cell, it would lead to the tissue damage.
Yes.
Okay, I'm have no question right now.
Thank you. So finally, the image of the picture of lupus patient is have more severity, right? From the molecular mimicry mechanism.
Yes, I do.
Okay.
Yeah, actually, I have one question, but I'm not sure we have time or not.
We have time, Asano. Go ahead.
Since the receptor for this virus is ACE2, right? And like Tamonwan already mentioned about coagulation, we know that COX or cyclooxygenase and ACE are involved in coagulation, right? So how can you relate this to? I mean, the you already mentioned about it, but do you have more information about this? Can you link the activation of it?
อาจารย์โอ mean the severity from the ACE2 receptor.
I mean the activation of the receptor because ACE or COX cyclooxygenase pathway is involved in coagulation, right? So the virus um, activate the receptor and can it link to the coagulation pathway in the patient? Is this clear?
May I have a question again?
Okay. ACE, right, is the receptor, I mean receptor for virus. And ACE is also involved in coagulation pathway. And the virus activate the receptor, right, in order to entry into the cell. And this activation, okay, is it involved in the activation of coagulation?
Once that our body get infected from this virus, so as a group one, as Ajahn says that they can increase the presence of ACE2, which is have like the functions in the coagulation pathway. So if this virus can do this, maybe they can lead like increasing the thrombosis pathway, not just only because of the antibody, not just because of the antibodies attached from the molecular mimicry. So maybe they can induce. So we may see the like the DIC more most in the patient too. I'm not sure this is that's the point that Ajahn Pasong said or not.
Yeah, I don't have the information, but I think it's very interesting because coagulation is occur because of the infection. Okay, but since this virus is very specific to ACE receptor, so that's the thing that maybe the researcher need to investigate more. But to uh, my knowledge, I don't have any information about this, but just curious about it.
Yeah. Can I share because I think in the article in article that they report the case they says this in this case they call COVID-19 coagulopathy that have one patient from 17 patients that they have the thrombolytic complication, Ajahn Oh. But then the mechanism that they don't mention what happened in the patients. But then there is a case, like I mentioned, one case from 17 patients that have the abnormality of the thrombosis complications. But then they don't mention about the mechanism, but that is the case. Yeah.
Yeah, and yes, and actually, group 3 gonna give more detail on the lab findings, right? So maybe we can hear from them again.
Okay.
อาจารย์คะ about the AACE2 receptors and coagulation pathway. Actually, I have one theory that I want to share. Actually, from my searching ACE2 receptor, they didn't appear only in the lungs. They can appear in many cells in our bodies, including GI tract or the vasculars or the brain. So that means a virus can infect these cells in in every part, especially in the vascular. If they infect in the vascular and lead like the injuries on the vessels, so they may lead increasing of the coagulation pathway and end up with a coagulopathy that Ajahn Pat said. So next, I will move to anti-phospholipid antibody syndrome. First of all,
in what mechanism? Right now, the first one is lupus patients have more susceptibility for the severity in the case of hypomaturation from the presentation. The second one is from the lost immunoregulation, that why lupus patients have more severity in case of COVID-19 infections. The third one is molecular mimicry, right? So
yes,
the next one is what? Antiphospholipid. This about what? In what mechanism?
is about in a mechanism of coagulation.
Possible mechanism when you
Yes.
present your friends.
Okay.
Antiphospholipid antibody syndrome. In what mechanism?
In the mechanism of loss tolerance.
The second one, right?
Yes, it is quite similar. So I will share the information from Mercury's. First of all, the anti-phospholipid antibody is described as an autoantibody that recognizes anionic charge phospholipid such as cardiolipins or the phospholipid binding protein cells such as beta-2 glycoprotein. In the normal conditions, the beta-2 glycoprotein, which is the main of the autoantigens, is a plasma protein that has an important role in the coagulations. But if they have like the second hit or the triggers that enhance develop severities in terms of the push the hemostatic balance in approval of the thrombosis. It might be increased to produce an anti-phospholipid antibody specific to the beta-2 glycoprotein. For about the second hit, they can including the infections, the inflammations, or the pregnant. Moreover, they have one fact that's a molecular mimicry between the structure of the bacteria or virus and beta-2 glycoprotein. They can also induce the anti-phospholipid antibody too. So about the mechanism of the anti-phospholipid antibody, once that they appear of the anti-phospholipid antibody and they binding to the beta-2 glycoprotein, it may this immune complex may be they binding into the toll-like receptor 2 for adenosine A2 or the LRPS, then they active the intracellular signaling pathway such as NF-kappa B, P38 or the MAPK, resulting in the induced pro-thrombotic phenotypes. And about the pro-inflammatory, for my searching, they show that most found in the first trimester of the pregnancy. Uh, in this point, they didn't clear why the infections they most appear they are appear pro-thrombotic phenotype more than the pro-inflammatory. So for all this, I will conclude that the infections, especially from the virus and also molecular mimicries, can be induced producing anti-phospholipid antibody and lead to the autoimmune.
So you mean that in COVID-19 patients, in severe case, they will produce anti-phospholipid autoantibody, right?
Yes.
And which one come first? Autoimmune disease first and then get COVID-19 infection to produce anti-phospholipid, or get infection first, produce anti-phospholipid, and that induce more severity in lupus patients. Which one? COVID-19 or SLE?
Actually, I think they can be the both of the way.
Explain us. If COVID-19 first, what happened?
If COVID-19 first, as I said before, that they can occur the apoptosis cells from the infections. So the apoptosis device, maybe they can be the phospholipid from the cell that dead, and these phospholipids maybe they can appear as the autoantigens if our body can our immune system they cannot recognize this is self antigens, maybe they can produce the antiphospholipid to target our antigen.
Do you have the evidence that COVID-19 patients produce anti-phospholipid antibody?
Yes. I sorry, first time I forgot to show the case. They have a case report that they appear the autoimmune disease once after infect COVID-19, and they show the antiphospholipid. They show the 3 patients from the China.
Okay. These are COVID-19 first, produce anti-phospholipid autoantibody that induce severe in lupus. What's about lupus first?
About the in SLE, actually they didn't appear only the auto antibody. Maybe in the underlying, maybe they also appear antiphospholipid already. But the second hit from the infections, maybe they can induce severities to turn to a patient in the SLE flare.
Yeah, thank you.
And and I will move to the next topic. Is about the immunosuppressive drug in SLE.
Yes. So for I will present that today is the last possible mechanism that can show the association between the autoimmune disease and the COVID-19. As you know, in the autoimmune patients, that in some like SLE flare, which has contributed like organ damage, so they need to use the immunosuppressive drugs for like the supplies or decrease of the immune system and to reduce about the effect from the disease on the patient body as well. And actually, the drug had like many types of the drugs, including like steroid or biologic or the anti-inflammatory as well. So next slide, please. So this one is the example of the drug, subversive drug. You can see the hydroxychloroquine, and I think that anyone in here maybe is heard about this drug. Is like anti-malaria drugs as well. But for the case this case, you can see the mechanism of the drug. HCQ is kind of like decrease of the T cell proliferation, TR activation decreasing, or decrease of the cytokine production. Also, it can reduce about the antibody production. As you seen in the figure, you will it can indicate that if this patient had like have the SLE for underlying disease and they are on drug treatment, that means the immune system of they will low, and then if got the COVID infection, that means it doesn't have the immune cell to be like agents to of fight to the COVID as well. So it can cause the serious symptom in the patients more than the normal patient or the patient without the autoimmune disease as well. And I have two more example for the drugs. For the next one is about Pina, can you go to the next slide? About the glucocorticoid or GC. From the figures, normally these GC are usually like used to combination with the HCG drug as well for be like synergies of the to get the good come out for the treatment in the patient as well. As you can see in the figures, like the more spherical in the violet color, combined with the steroid binding protein, and then bring the GZ come part to the cell, and then the activate the receptor. GR is the GZ receptor as well, and the complex can directly bind with the transcription factors about the pro-inflammatory genes, and then it cannot occur after binding directly binding with the factors. It cannot occur about the transcription, translation, and explosions of the pro-inflammatory cytokine, and that means it doesn't have the cytokine to be respond for the virus infection as well. Yes, and another one is about the biological agents, or I can say that about the targeted drugs as well. As you can see in the figures, it has like many drugs that can inhibit like molecular in the surface of B cell or the molecule on the T cell as well. For example, in the B cell, you can see at the plasma cell, the plasma cell is this can produce the antibody as well, and you can see the drug bortezomib can inhibit the proteasome on the plasma cells, so this can cause like the plasma cell cannot produce the antibody to agents for the virus infection as well. And actually in the T cell as well, like tofacitinib in it can inhibit the JAK-STAT pathway also, and that it cannot send the signaling to the nucleus in the T cell and resulting in the downregulate of the T cell. So from this possible mechanism, we need to consider, because if at first the patient had the SLE for the underlying disease and then got the drug for treatment and got the low immunity or immune system in their body, and it means that if they got the infection from the COVID, that means they can be have like a severe symptom, and as well maybe they could die more like I had high potency to cause to occur the severe symptoms than the uh, other patient does not infect about the virus as well. Yes. Thank you for your kind attention.
More question from student, question, discussion, or any point that you are unclear? Make it clear for now.
I have some questions.
Oh,
Go ahead. Yep.
แค่ทวิตเสร็จแล้วก่อน you first.
I curious on the immunosuppressed drug part. You say that in the patients that use the immunosuppression drug, you have more severe symptoms than the normal patients, right? So, I wonder why they have more severe, because from the group 1 presentations, the severe symptom of the COVID-19 is come from the cytokine storm that is about the hyperinflammation. Also, I think if the patient use the immunosuppressed drug that can decrease the cytokine, should it be prevent the cytokine storm or prevent the symptom of the patients?
So cute. I think this question is so cute. Okay, answer it.
Okay, at first that I said that it had like many type of the immunosuppressive drug. It had like anti-inflammatory or the targeted one. And if you like use the anti-inflammatory in the patients, maybe it can cause not much at like not much the cytokine storm if they got infection from the viral, but it still had like another cell for T cell, maybe T cell it can like to cause the directly respond to the virus as well. But the more severity doesn't mean it's come from like when okay from this one HCG, you can see that if these patients are on this drug, you can see is T cell proliferation is decreased, cytokines decreased also, and antibody is also you can see is like over almost cover all the immune cell that can decrease. I mean, it can cause you have the low immunity. So when you have like no cell to be fight with the virus and then got the virus, so it can cause like virus can stay in your body like freely. I don't have any cell to get rid of me or anything else. So it can like infect and then transfer to another organ to cause the inflammation in the body organ as well. So that can cause the severity symptoms that I mean. Yes.
But how Machima, how it linked to the autoimmune disease?
You mean the drug using?
I mean the effect of the drugs.
The effect of the drug can be good for the patient that have the autoimmune disease because it can reduce or decrease of the immune system to be like it don't have the auto antibody to bind with the cell antigens in the patient, so it's not called like uh, I mean this can be improve their symptom as well. Yes, for using the
You mean that if the patient has an autoimmune disease and then treated with the hydroxychloroquine and then subject to the viral infection, you
Right.
mean?
Yep.
yes.
Have you heard in another way, Rao? Because there was the idea to use this hydroxychloroquine to treat the COVID patient as well, right?
Yes. I found the papers that show this drug can be like treated in the COVID-19 as well. In this figure, you can see a decrease of the cytokine production in the interleukin 6, especially in the interleukin 6. At the group 1% that the interleukin 6 is a significant highly amount of the cytokine from the COVID infection. So if the patient that on treat with the HCQ, that means and they get the COVID as well. That means this drug can also improve the symptom in the SLE and also can reduce the interleukin 6 that can be like cause the inflammation more in the COVID-19 infection as well.
So, but would it be possible that if the patient, the COVID patient, never have a autoimmune disease and then treated with HCQ and then turn to be an autoimmune disease?
You mean again, please? Again, please.
Just another way around, because when you present, you propose that the autoimmune disease treated with hydroxychloroquine, so it suppresses the immune system, and then the autoimmune disease patient susceptible to COVID infection. That's what you propose, right?
Yes.
Yeah. So I just asked in another way around because they use this HCQ also to treat the COVID patient. Would it be possible? Yeah, if the COVID patient will turn to the autoimmune disease, what do you think?
You mean
Sorry, Achanya, you mean the patient is autoimmune already, right?
No.
It just non-fair or no? No,
No.
like us, get COVID-19, right? And then treatment with the HCQ. How is possible to turn to autoimmune disease, right?
Yeah.
Okay. Yep. I clear that question. Yin, answer your question.
Just for my opinion,
it is possible. If yes, yes. If no, why? Nobody know actually. This just you share your idea.
For me, I think maybe it's possible because I thought that if you're using this drug for the long term, it can affect to the kidney, like the complication in the autoimmune as well. But if at first you don't have any factors or like any hereditary genes as well, the genes from the families. So maybe if you use for the long term, maybe it can first of all cause the effect to our body, but
you not call autoimmune disease, right? It's just the symptom that similar, but you not call autoimmune disease. That's what I guess from you. I buy your idea, but I still don't have the right answer. Yeah, but I buy your idea.
Yeah, so I hear her from looking at this information. So actually rheumatologists just worry about using HCQ for the treatment for their patients. I mean autoimmune disease, right? Because HCQ is if you saw the news, it seems like the withdrawal, right, Ajahn Chaniya? Try to withdraw HCQ, that then rheumatologists that worry that, oh, how can they treat the patient because HCQ is cheap, you know, because it's cheap. So then the doctor is easy to treat them, but then if you withdraw them, so then what happened? So then they worry about take care of the patient. So this is a problem from the clinicians. So clinician talking about how they can manage the autoimmune disease patient that it need come to follow up all the times. So then it seems like disagreement for using HCQ in the autoimmune disease. I mean, in fair patients that have symptoms. Yeah. Yep. But for the question that how can HCQ induce autoimmune disease, I think it's unclear and no data so far to make it clear. But it's just the point that need to worry and concern that possible. Yeah. Any questions before we move to the last one?
Yes, I have a question.
Yep.
Please forgive me if I'm wrong.
No,
I want
okay. Yeah.
like molecular mimicry. While I was reading, I found some other mechanisms that was release of twisted antigen ectopredin and exposure of cryptic cells, uh, uh, epitopes and breakdown of cell energy, ANERGY. Uh, just like molecular mimicry when I was reading for this topic. Well, did you have any idea about this thing? Because I was not able to get much information about these mechanisms and all. They just gave a definition of everything.
Yep. So what's about give the definition to your friend first?
No,
Nine.
No,
Molecular
actually,
mimicry.
yeah, like just like molecular mimicry, the other mechanisms which I came across, that is epitope spreading in exposure of cryptic self epitopes, then breakdown of cell energy and release of twisted antigens. But I don't have much idea about that since it is their topic and while fine studying for that, did they came across these things, these mechanisms?
Long Nai, can you have the slide about the showing the viral antigen?
Viral antigen here.
Yeah. To explain similar about the molecular mimicry.
Not more. It's actually these are separate mechanisms, just like molecular mimicry.
But
So
Chimika, you just want to know what is the sequestered antigen, right?
yeah,
Because that's one of the mechanism of the autoimmune
yeah.
disease, which
In
is nothing related to SARS-CoV, but you just want to know what is the secreted antigen and how it comes
when
from.
I came to know about this when I was reading for this SLE and sarcovy. That's why I'm asking. I don't
Mm.
know much about that. When I was reading for this papers, this assignment, I came to know about these things. And I thought the SLE group people will present in detail.
Mm. Sequester antigen, maybe I can help. Or Tamon, what you want to try?
เขาถามว่าอะไรนะคะ
She asked if she understand well about molecular mimicry already. But then another possible mechanism of autoimmune disease is the sequestered antigen. Yeah. So she's not so clear about this, and this sequestered antigen is related to SARS-CoV or not. That's what you asked, right?
Yeah, yeah. And not only that, and also there is epitope spreading in exposure of cryptic cells epitopes.
Mm.
This I just came across this. I don't know much about this, and I thought just ask whether this is related to this or not.
I don't think I have the data.
Mm. So
And
then maybe I
it's
can
okay.
try if I understand you correct, Chimila. Yeah, because sequester
Yeah, nobody
antigen,
understands me.
the sequester antigen, it means that the antigen in our body or in any part of this world that our immune never seen it before. When our immune develop, so in order to not to harm to our self antigen, it means that our immune system need to get rid of that clone that seen this auto antigen before. But anyhow, not every antigen in our body that the immune system have seen. This what we call secretor antigen, which is means the antigen that hiding in somewhere that our immune never seen. The example of this one is would be the retinal antigen in our eyes or even the cement. Yeah, this thing is kind of the antigen that our immune never seen. For some reason, if this antigen break out by the accident or something that trigger, so then our immune cell will see this antigen as the foreign, not our self antigen anymore. Yeah. So then it start to attack, and this antigen is on our cell in our body. That's why it's called autoimmune disease. Yeah, that one thing. So linked to the SARS-CoV. So I don't think that the secreted antigen will related to SARS-CoV because SARS-CoV is something that I think it would probably fit to the molecular mimicry. Yeah. So our immune system see the SARS-CoV antigen. Unfortunately, that SARS-CoV antigen is the same as our self antigen. Then all the cells that get activated react to self antigen. Am I correct? Or this is answer?
I don't know. I don't know. I just came across this. I also didn't get much information about this in the epitope spreading. There are 4-5 mechanisms. I don't have much information about this. I just asked whether this was
I actually have
related.
one thing to share. Like Tamon already mentioned about anti-phospholipid, right? And when it's damaged because of infection, okay, the antigen, especially the inner how you call it, inner membrane of the cell, right? It exposed. That means now you can see the antigen that you have never seen it before, right? Especially like cardiolipin and other phospholipid, right? So that's the thing, part of the self and cell antigen or sequestered antigen that you have ever seen before. You have never seen before, but once you get infected and the cell get damaged, then it's exposed so that your immune response can see it. So that I think it's one of the example.
Yes, but if I think Ajahn Tia is correct because in the way of COVID-19, I don't think a secretor antigen is involved with this one from my searching. Molecular mimicry is just like cross-reactivity that they mentioned in this, actually cross-reactivity. So anti-phospholipid autoantibody it's just one part of molecular mimicry. But they say like anti-phospholipid autoantibody it's not involved with molecular mimicry at all. They also say like that, like in lupus patients produce anti-phospholipid autoantibody, and that autoantibody can induce the severity in COVID-19 patients. So it just use associate, but they don't know what mechanism that could induce.
Yeah.
So
I
yeah.
think it's not molecular mimicry, but it's the sequestered antigen. Okay, that your body have never seen it before. But when the cell get damaged, yeah, it's exposed. Okay, but it doesn't mean that it's molecular mimicry. It's just one thing that hidden in your body, inside your body.
Yeah. Thank you, Ajahn. Oh, Ajahn Shaniya.
Okay.
More question from students before we move to the last question or discussion or sharing the knowledge.
I just wonder about the treatment of the autoimmune patient when infected with the COVID-19. Is meaning that that patient must stop using the immunosuppressive drug to treat the COVID-19 rate, or is not necessary to stop using it.
Okay, I will try to answer your question, but I quite know the exact like solutions to this problem. From my opinions, I think that they should not stop using the immunosuppressive drugs, but they should try to find another way, like combination with another drug to reduce like some severe symptoms. Because maybe one drug can directly to uh, buy or target on the one thing and cause the one outcome, and another one is can be like reduce something that can be, I mean that if we combine together maybe it can reduce in some severe complications and the patients. But if the patient stop to use the drug, that means the autoimmune disease, I mean on underlying disease of them maybe like still have and can cause the affect also, and it doesn't good way to be stop to use it, I think. Ajahn, can you add me more?
Ajahn, it seems like this, like you are clinician and you have the autoimmune patients that you need to take care of them. The patient is, need to come to the hospital all the time. And then the challenge of clinician is how to suppress to non-fair patient, right? But then once happen, they come to the hospital or to the way, and then they get infection with the COVID-19. If you are a clinician, you would like to treat COVID-19 first or lupus first. You have no choice. That's why rheumatologists point up this. Please help me. What is the guideline for rheumatologists, right? So then If you are a clinician, you would like to treat COVID-19 first, or you treat lupus first? Which one?
I think COVID first
Okay,
because this I think to treat the lupus SLE is harder and take more time. So if we change to treat the COVID-19 that can be killed faster, maybe is better way.
even me, I don't know because but if I'm clinician, I think I gonna treat COVID-19 first to stop something. Then if they feel better, or I can suppress cytokine storm or whatever, then the manage of the lupus patients will come if I am clinicians. So that's why in the time of the epidemic, the clinician just ask lupus patients stay home, don't come to the hospital. Seriously, stay home, don't come to the hospital. They just delivery drug to the home, just quarantine themselves. So they ask the patient, don't come to the hospital. Be careful. Please try to be safe, not infection. If not infections, that is the most difficult to treat for lupus patients. Even in our country, the doctor do like that. Yeah.
For more information, for treatment of the COVID-19, they use the combined drug, the antiviral agent plus the immunosuppressive drug, especially IL-6, right, that one. So I think, as Ajahn Pachani mentioned, that for the patient that have the underlying disease, any underlying disease should be safe from the infection, because the treatment quite very complicated, that one. So for Mashima, I think you already present about the immunosuppressive drug, right? But in the real situation, they use like antiviral drug as well. Like they try to use the anti-HIV, HIV drug to suppress the virus, because they use the same enzyme for replication. That one.
I have some question connect to the Ajahn said before. If use the HIV drug, it can be more CD4, right? It can be more cytokine storm and can be the severe COVID-19.
Good question. Ajahn Onaritai, help me, please.
Why use anti-HIV drug have more CD4?
This patient has no HIV infection.
Yeah. So they stop the virus to infect the cell, right? Any cell, any target cell.
Yes.
So I think the lymphocyte will go to the normal level if we can treat that one, but it not go too high, like you said.
Yes. Thank you very much.
อาจารย์โอคะ could I a little bit about the using anti HIV drug compare with the hydroxychloroquine which inhibit the terminal glycosylations of ACE2. Which one is better for the treatment COVID-19?
I'm not sure, but for my review, SCQ is not used anymore. Like you see the Donald Trump try to encourage the US people to use SCQ for protection, but it's not in that case. So I think compare between HIV drug and SCQ, the anti-HIV will maybe more efficient, because they can kill the virus directly. They can inhibit the virus propagation, right? So if there's no virus in the first place, there's cell attachment, something like that.
Yeah, it's interesting, right? So that's why I give this topic to you guys because very new and not much knowledge. So if you can answer, that means you bring your basic knowledge, come to update with the news, with the new things that coming, So that's, I think we should done with the question this one. We move to the last question that, you are empty. So then you should having fun with the lab test that, I give you the package of the lab test. All of that is empty professional. Then you need to integrate and tell us what happened in the patients. And Ajahn Opasong will take care this question. Right. Yep, go ahead.
Okay, everyone, our group will present what's the laboratory finding in correlation with pathogenesis and clinical manifestation. So next slide, please. First of all, when we look at the lab finding, we have to find the abnormal lab result, right? And this patient had a sign of the lung abnormality when using imaging technique, which is CT scan. It appeared bilateral patchy shadowing for blood. And for the blood routine test, the lymphocyte and the hemoglobin are lower than the left range, and the D-dimer suspect are high, and this patient has high D-dimer. They suspect the patient might have the hypercoagulable condition. So next slide, please. And when we have to look in the detail of lymphocyte, the helper T cell and NK cell are decreased. And the serology test, we can interpret the result that the patient doesn't meet the criteria of the SLE condition, but this patient may have the systemic autoimmune underlying condition. And this patient is infected by the SARS-CoV-2 virus. Next slide, please. For the blood chemical test, it appears low level of albumin and high level of lactate dehydrogenase, which indicating the tissue damage. Next slide, please. For the infected related biomarker, there are three pro-inflammatory cytokine are increased, which are interleukin-2 receptor, interleukin-6, and tumor necrosis factor alpha. And for all the inflammatory cytokine are increased. Next slide, please. So we will start from the chest CT scan and see what happened in his lung and try to connect the clinical manifestation, cytokine storm, pulmonary embolism, and deep thrombosis with the abnormal lab result as we described in previous slide. So next slide, please. From this chest X-ray testing, the result show the bilateral patchy shadowing, right? Which is the sign of the acute respiratory distress syndrome, or ARDS. This syndrome can caused by many factors such as sepsis, inhalation of harmful substances, severe pneumonia. But due to the pandemic spreading of the COVID-19, it is the most suspected cause of the acute respiratory distress syndrome. And we also confirmed by liver transcripted PCR. Since SARS-CoV-2 entered the alveolar epithelium through the ACE2 receptor, the immune cell, including macrophages, neutrophil, and monocyte that respond to the virus, will travel to the site of the infection and produce cytokine and chemokine at the site of the infection and cause the damage to the protective membrane and makes the fluid leak out from the blood vessel into the lung, into the alveoli. And the sign and symptom of acute respiratory distress syndrome will be like shortness of breath and rapid breathing because there is fluid contained in the alveoli space, and unable to access the same oxygen level as normal. So our body will try to compensate by increase the respiratory rate. And so Shimila will explain more the cytokine and the chemokine that inflammatory cell secrete. Shimila, are you there?
Hub, you need to present because she doesn't hear you, I think. You call her?
Sorry. My God. Okay. Among the, I'm very sorry. Among the abnormal laboratory findings, I'm going to talk about infection-related biomarker cytokines and correlate them with clinical manifestation and pathogenesis. As you can see from the table, the one of the most important pro-inflammatory marker, which is interleukin 6 and tumor necrosis factor alpha, have been elevated in this since this patient is COVID positive. As soon as virus enter the body, the once virus enter the body, the virus invades the respiratory epithelial cells. In starting from that point, the first line of defense mechanism, this is the innate immune response will be activated. During an innate immune response, the macrophage, monocyte, dendritic cells, and epithelial cell, these are the major source of cytokines in the cytokine which they release supposed to give a protective response in it should protect the body from the virus. But in case of SARS coronavirus, what happens is the exaggerated response of the immune system causes the disease. This is the main reason for this is the release of cytokines, large amount of cytokines. As you can see from this slide, initially when the infection occurs, there is very limited amount of cytokines in some few chemokines. But when the amount of cytokine continues to release for non-stop for large number, what happens is it led to the cytokine storm. And once the cytokine storm will occur, as a result, the cytokine results in influx of various immune cells such as neutrophil, macrophage, T cell at the site of infection from the circulation, and this results in hyperinflammation of the infected site. And initially, this is the lung, and the lung infection is one of the very basic initial finding of the cytokine storm, and it can be multiple organ. And the consequences of the lung infection will vary from acute lung infection to um, acquired respiratory distress syndrome. In acquired respiratory distress syndrome, which is a severe condition, in acquired respiratory distress syndrome leading to low oxygen situation level is the major cause of death in COVID-19 also. In next slide, please. And as you can see from this picture, and this is the pathological effect of cytokine storm on human tissue. As you can see from this, due to the excess release of cytokine, as a result of hyperinflammatory reaction, there is a destabilization of endothelial cell-to-cell interaction, and the vascular membrane is damaged, and there is formation of hyaline membrane. You can see from the pink color the hyaline membrane, and the vascular membrane leakage also clearly. I'm sorry, I don't have a pointer. In the protein-rich edema fluid accumulation in the lung, in overall there will be a diffuse alveolar damage. This is just to show how the lung will be, the alveoli of the lung will be after hyperinflammatory reaction. Next slide, please. In the cytokine storm, which is not present in all the patients, and it only happens in severe and fetal cases. In the cytokine storm is characterized by clinical presentation of overwhelming systemic inflammation, hyperphosphatemia, hemodynamic instability, and multi-organ failure, and if left untreated, death may occur. In the trigger for cytokine storm is uncontrolled immune response. In the cytokine storm, which is seen in SARS coronavirus, is almost similar to the previously masked and SARS virus, and also the same cytokine response will be seen in the cancer patients who are receiving chimeric T antigen receptors. And among all the cytokines, the most they have labeled interleukin 6 as the culprit. In interleukin 6 is the most frequently associated, and it is considered as the prognosticator for the patient with COVID-19. And most of the retrospective studies regarding cytokines reveal that high level of interleukin 6 is associated with mortality. So I will just go a little bit detailed. This picture has already been presented, but I will just give little bit detail about how it is. As you can see from cytokine, the interleukin, the cytokine will, among cytokine, the interleukin, basically it will function through two pathways, that is cis signaling and trans signaling. When it signals through the cis signaling pathway, what happens is it will have a pleiotropic effect. Pleiotropic effect means it will enhance both cell mediated immunity, and also it will innate and acquired immunity. That means the interleukin will have effect on the T lymphocytes. And one of the main features you can see from this is the interleukin 6 will will decrease the amount of T regulator cell. And also it will help to increase the T helper cell 70, and T helper cell 70 is the cell which has almost same effect as interleukin 6. When the interleukin 6 is activated by trans-signaling pathway, what will happen is the endothelial cells and the epithelial membranes which do not have receptors for interleukin 6, it will make that cells to upregulate interleukin. And the effect of that will be there will be secretion of there will be severely exaggerated cytokine storm. And this is and it will cause the release of vascular growth factor and macrophage chemoattractive protein, interleukin 8, and also interleukin 6. And the main feature of this is one of the main protein which will maintain the vascular integrity is E-cardenine, and it will reduce the amount of E-cardenine, and this is something which is related to the fluid edema. And it will alter vascular permeability and leakage, which participate in pathophysiology of hypertension and pulmonary dysfunction in acute respiratory distress syndrome. And the other thing about interleukin 6 is without any pathway also. Interleukin 6 directly travel to the liver, and it will cause increase of CRP, uh, heptazidin, fibrinogen, in thyropoietin, and complement 3 receptor ferritin, and it will decrease the level of albumin. And this effect it will have without any involvement of pathway. The interleukin which is saturated directly travel to the liver, and it will have this effect on in the high level of CRP in fibrinogen, which will be seen in the COVID-19 patients. This is one of the main reasons. In the other inflammatory, the other one which is seen is tumor necrosis factor alpha. In tumor necrosis factor alpha is inversely related to the T cell count. As soon as the COVID-19 infection progresses, the tumor necrosis factor alpha will increase, but the lymphocyte count will decrease. When I was searching for this, I came to know that SARS-CoV-2 spike protein induces tumor necrosis factor alpha converting enzyme CAAC dependent shedding of the extracellular um, a- angiotensin converting enzyme to receptor domain, result in the loss of AG2 function and production of tumor necrosis factor alpha. That means whenever the virus will get infected and the infected new cells, there will be release of tumor necrosis factor alpha without any involvement of immune cells. In the other abnormal finding among interleukin is interleukin receptor 2, interleukin 2 receptor. Actually, normally in COVID-19 patients, the amount of interleukin 2 will be low. Interleukin 2 will act as a growth factor for growth regulation factor for T lymphocytes. But in this case, interleukin 2 receptors are more. The main reason for the increased amount of receptors is the uh, interleukin 2 receptors are present in only lymphocytes, and it is separated by activated T cells, and elevated T cells are used to marker for T cell activity to other inflammatory process. What happens is whenever there is the T cells are activated, and when they do any function, whenever they get accelerated, and there will be shedding of interleukin 2 receptors into peripheral blood. And this directly correlates with the activity of the T cell and activity of the inflammation. Next slide, please. In the since I'm present in the cytokines, among all the cytokines, the clinically important one is interleukin 6. Interleukin, the main clinical features of interleukin 6 is interleukin 6 can lead to the vascular leakage, activation of complement and coagulation, and also it led to the symptom of cytokine storm, such as disseminated intravascular coagulation, cardiomyopathy leading to myocardial dysfunction, activation of endothelial cell leading to capillary leakage, hypertension, coagulopathy, and also it will lead to the kidney altered kidney function, anemia, and liver damage. In the other thing which I found about interleukin 6 is the most of the cell receptors, even in the kidney, heart, brain, the interleukin 6 will have effect on this. The interleukin 6 will make the kidney cells and the cardiac cells to take the virus. And what happens, and this is proven by the deceased pathological finding from the deceased person. And the other one is tumor necrosis factor alpha. The tumor necrosis factor alpha will induce a flu-like symptom with fever, general malaise, and fatigue, but can cause vascular leakage, cardiopathy, lung injury, and activate acute phase reactant, acute phase protein synthesis. And the acute phase protein synthesis part will be taken by the other person. I'm sorry, I don't know the name.
Okay. Since COVID-19 can induce interleukin 6 productions and cytokine storms, increasing in these pro-inflammatory cytokines, especially for interleukin 6, can affect the acute phase protein productions in hepatocytes. Interleukin 6 interleukin receptor complex will bind to the GP1V0 on the hepatocytes, and signaling is initiated through activation of the intracellular signaling pathway in hepatocyte. This signaling pathway leads to activations of nuclear transcription factor, consequently increased productions of positive active phase protein and decreased productions of negative active phase proteins. Positive active phase protein is the protein that increases in plasma concentrations in response to inflammation, and negative active phase protein is the protein that decreases in plasma concentrations in inflammations. For, for the positive active phase proteins, uh, there are CRP fibrinogens, haptoglobin, and procalcitonin. CRP is a protein that plays a role in innate immunity by enhancing complement cascade and inductions of cytokines. Next is fibrinogens. High level of fibrinogens in the blood can cause to increase the blood viscosity and promote clot, which may lead to hypertension. And high fibrinogens also can make liver cells stick to each other more easily, which settle faster and result in high erythrocyte sedimentation rate. For the haptoglobin function is used to clear free hemoglobin from circulation. High haptoglobin lead to decreased free hemoglobin and limiting hemoglobin ion available for the pathogen. Last is procalcitonin. Procalcitonin in COVID-19 is a widely used biomarker to assess the risk of bacterial co-infections and disease progressions. Because procalcitonin is a sensitive marker of bacterial infections and also respond to the tissue injury and inflammations. High procalcitonin in COVID-19 can use to suspect that the patient has acquired bacteria or his symptoms become much worse and go into drugs or develop an inflammatory syndrome. Like this. For the next negative acute phase proteins, there are albumin and transferase. In inflammations, liver reduce productions of albumin to allow greater increase to increase the amount of amino acid available for positive acute phase protein productions, and reduction in albumin may also caused by inflammations that increase the vascular permeability. Uh, decrease, Decreasing in transference can lead to reductions of the ion transportations and limiting the ion availability for the virus, and also lead to increase in ferritin. Inflammations also can induce hepcidin productions in the hepatocytes. Hepcidin is the regulator of the ion's metabolism that interact with the ferroproteins. When the hepcidin will bind to the ferroproteins and limit the ion's availability for the virus.
For the next, I will try to link the COVID-19 to the cytokine storm that cytokines that COVID-19 before group can present that and occur the cytokine storm, especially interferon 6, which is a predominant in COVID-19, also cause activation of the coagulation pathway by increasing the sum of the coagulation factor. This leads to the uh, hypercoagulable state. In the severe case, it occur the such as the pulmonary embolism or the DIC. And also the cytokines also activate the megalopoiesis and increase the platelet and increase the amount of the platelet and uh, uh, and some paper they also tell about the fibrinolysis shutdown, which is the decrease of activity of the degrading of fibrin caused to fibrin degradation product or FDP, and another cause of the D-dimer high is the autoimmune. They may have anti-phospholipid antibody, which is the antibody against the cell membrane. It causes the injury of the endothelial cell and lead to the activate platelet adhesion and platelet aggregation as well. So this is possible the cause to get the D-dimer high in the result. And another possible mechanism of the increased coagulation is the uh, renin-angiotensin system. Normally, they are the ACE2 to convert the angiotensin 1 to other form of the angiotensin 2. And so angiotensin 2 availability is decreased. That act as the increased coagulation. This mechanism control the regular level and angiotensin 2, but the COVID-19 makes the ACE2 decrease because COVID-19 competitively bind to the receptor on the epithelial cell lead to cannot produce ACE2 enough for moderate level of the angiotensin 2. When angiotensin 2 increase, the coagulation is activated more than normal. This is one of the reasons may lead to the COVID-19 patient have a hypercoagulable state to get the such a primary embolism or the DICs.
Okay, now I will continue into the lymphocyte part or the cell part. Because of the lymphocyte, which is in the white blood cell count, is in the skinning in the CBC test, because the CBC parameter is basically when we have the patient in the hospital. So when we have the CBC test, the white blood cell is normal, but the lymphocyte is quite low. As you can see, the result is 0.4, which the reference rate is 1.1. So it's quite low, and maybe which another laboratory parameter will have the significant, we can diagnose it as a COVID-19. So we go to the total lymphocyte count, which have the abnormality in the helper T cell and NK cell as well. It's quite low, which is the significant in the study now that COVID-19 patient may have the lymphocytopenia. Next slide, please. Because of the cytokine storm, which Shimila says that, and group 1 said in the COVID-19 patient. So they have cytokine storm that can lead to the lymphocytopenia, which is the reduced lymphopoiesis, induced apoptosis, and T cell exhaustion, and recruit a lot of lymphocyte to the damaged tissue. In this picture, which will clarify that out info topic: impact T cell depletion and exhaustion, lymphopenia, and T cell impair and dysfunction. First picture is impact T cell is caused by ACE2 receptor expression on lymphocyte or the T cell that can promote the SARS-CoV-2 into the lymphocyte and can cause the impact T cell. And the second is the T cell depletion and exhaustion. Increase in inflammatory cytokines level promote to depletion and exhaustion of T cell. And the third part is viral infection, which is the SARS-CoV-2, directly damage the lymphatic organs such as the spleen or the lymph node. So it can cause the lymphocytopenia. And the last one is the inflammatory can cause the lactic acid level. So the lactic acid level can inhibit the proliferation and the dysfunction of the lymphocyte. And so all of these can cause to the lymphocytopenia and make the CBC parameter is abnormality. And we can screening or we can diagnose it as the COVID-19 patient.
May I comment first before ask the question for this group? So you come in general knowledge of the lab finding, but then you don't tell us what happened in this patient. So can you summarize to your friends and to our staff what happened in this patient from the lab finding? What happened from the lab? You come like blah 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 blah. This lab maybe, but what happened in this patient? Can you explain? Can you summarize? Too much information for me a lot, but it's good to give your friends to basic, and then for bring up this knowledge to the knowledge, and then give you the assignment 2 that you're gonna work for 2 weeks, and then tell us for the in October. But can you summarize what happened in the patient for these patients from the lab finding? I mean, what happened in the immunogenesis or immunopathogenesis? What happened in the patients?
Go to the slide 6, please.
Yeah, just summarize, like, this patient is get COVID-19 and have what pathogenesis?
First, the first evidence is a bilateral patchy shadowing, which reflects the ARDS syndrome, and then we suspect that COVID-19 may cause that syndrome, and we get the result of the RT-PCR to confirm that this patient has infected by COVID-19 patient, and then we gonna guess that what caused the ARDS So we suspect the cytokine storm, and when you look at the cytokine storm, we can prove with the lab, which is interleukin 2, interleukin 6, or TNF alpha, which is play main role to the cytokine storm. And then the result appear that the interleukin 6 and TNF alpha are increased, and when we try to and we saw the D-dimer result, that we try to correlate that what's going to happen, and we found the evidence that interleukin 6 may activate some coagulability to may improve the pulmonary embolism, prevent thrombosis, or something else. That evidence show that the dimer has increased. So, and the consequence of the cytokine storm, they get the inflammation, and we can detect the consequence of inflammation by acute phase protein such as CRP, ESR, ferritin, albumin also. This is the overall.
What about anti-double strand DNA and ANA? What you forgot them at the hospital?
I'm not forgot that, but I think in this patient it didn't have the SLE condition, because in the criteria of the SLE, they should have the titer more than 1/80.
So you just exclude out no autoimmune disease in this patient? Completely.
I said that this patient has systemic autoimmune condition, but not SLE.
Okay, and then you don't add up for the pathogenesis at all to explain the lab?
For the pathogenesis, can you go to the slide of the coagulability?
Actually, the slide overview is really perfect, but how can you add the autoimmune condition? Okay, we call in condition, right? In that slide, in the overview, how
can I add it?
Yeah.
I will add up in the, I think about the cytokine storm, because the autoimmune is the antibody against the cell antigen, that it may occur the some inflammatory event in the body. Yes. This is my just thinking.
Then you think this patient is just same like us, healthy patient get COVID-19 and then produce anti-double strand DNA and ANA? Or you think they have underlying autoimmune condition with the ANA and anti-double strand DNA and get COVID-19? How can you link the knowledge pathogenesis from the first group, second group to explain the lab that this is a real one? This is the real case that happened.
Maybe after the COVID-19 infected, and they had some molecular mimicry and produce some autoantibody and makes the double ANA positive, but not in a high titer.
Okay, COVID first, and then induce ANA and anti-double strand DNA. Yeah, it's possible. Okay, Chimala, you don't think like half, right? Share with us.
Because ANA and DNA are very specific. It is anti-nuclear antibody and anti-DNA antibody. And COVID-19 will lead to the hyperinflammatory reaction, and hyperinflammation will cause the synthesis of acute phase reactant proteins. The acute phase reactant proteins will not have any role with anti-nuclear antibody and anti-DNA antibody. This patient actually has underlying autoimmune disease, but it's not SLE. So we didn't focus much about any other condition. We were just focusing on COVID.
Yeah, I get it. Underlying an autoimmune condition, you think, and autoantibody can go for
And
inflammation.
the main reason what I'm saying this is among the 17 patients which results have been presented in the research paper also, they have presented interleukin receptor 2 was decreased in 2 patients. And when I was reading for this interleukin 2 receptor, I came to know that in systemic lupus erythematosus, the interleukin receptor gene is mutated. In the presentation of interleukin 2 receptor in systemic lupus erythematosus patient is not significant. In this paper, it is specifically they have indicated 2 patients among 17 patients which they have taken for this study. 2 patients have decreased interleukin 2 receptor activity. But this patient don't have SLE because if SLE means interleukin 2 receptor will not be high, because in SLE that is interleukin 2 is decreased, gene is mutated.
Okay.
How do you know it's not SLE? Because maybe SLE and inactive. So that's why I give you just low titer, maybe they on treatment and then suppress it and then low titer. How can
No,
you
I was
how can you
I
make sure
was
like no SLE?
because interleukin 2 receptor, when I was reading for interleukin 2 receptor, I came to know that in SLE patient, interleukin 2 receptor is mutated. The amount of interleukin 2 receptor in SLE patient is low.
Uh-huh. Okay.
So that is how I rolled out it.
Yeah.
And I have evidence for it.
I mean, you just go to the point of L2 receptor, but you just think that maybe patients, there are many factors, to induce autoimmune conditions. But
Yes.
just be careful that not just only cytokines, one cytokine increase, and then you say
No, but I believe in molecular biology results so much.
Okay.
Because this gene mutation is a molecular biology, and being a lab tech, I think we just not depend on any biochemical. When it comes to molecular means, we will trust more. In everything confirmatory stage, we will go to molecular testing.
Okay, so I will add more lab finding and then discuss for that. But one thing from HAB is don't say that antibody is low and then no SLE. Maybe just inactive SLE. So then, on the condition of autoimmune disease. But for now, maybe just, no SLE, no active SLE. Okay. Ajahn Oh, sorry, you can go ahead.
Oh, that's okay, Kap. Yeah, actually the story about COVID-19 is quite good, but yeah, I agree with Ajahn Patani that you miss the underlying disease, right? Because now we don't know this phenomenon is caused by COVID-19 or caused by the underlying disease, right? So one thing that you have to differentiate. So far, we don't have idea about that. Right?
Yes, but then I want to see again from lab finding. So then what this patient is severe, non-severe COVID-19? Can you do like differentiation from the lab and then this is severe or non-severe or mild from the lab finding?
This patient is not severe.
Why? For what lab?
Because
That you can say that.
Among the laboratory findings presented here, actually this patient is not severe. It is actually mild.
But in order to say that the patient has flare or not, right? How can you do that, right? Can you find any index or indicator to say that the patient is flare up or not? I mean, the autoimmune disease in the patient is flare up or not? So can you find any index to conclude that the patient
maybe in the coagulation part, in this patient has the dimer high only, but and PT and APTT in a normal level. It can reflect the progression of the patient.
Yeah, but back to Ajahn Pachani's suggestion, right? If patient have already underlying disease with SLE. Okay, but if you want to follow up the patient, okay, can you find any index? Can you suggest any? I can say like the scoring system, okay, to tell you patient have flare or not.
I think the part of the information maybe can monitor of the severity of this patient.
Which
But
one? What marker? MT. Come on. Oh, which one?
in the marker of the information. Yeah.
Which one? Yeah, you
Which
say
one?
information. Which one? If you would like to monitoring or you want to say, this
Uh,
rare patient or remission patient with my COVID-19, something like that. What marker? If you work in the lab. Huh? Don't say like inflammation. Oh no. What biomarker?
Maybe C-reactive protein.
CRP? Okay. Why? Only CRP. CRP high? Oh.
No. But the first one that we use to see the inflammatory in the inflammation in the patient is the CRP, the C-reactive protein. Uh,
Here, C-reactive
อาจารย์คะ
protein is not specific. It is a acute phase reaction protein, positive acute phase reaction protein. It will be high in any condition, not only cold. CRP and ESR is a non-specific indicator. We can
You
just
see
conclude based on CRP.
disagreement in the class. So okay, what else?
Excuse me, could I add up in terms of the criteria in the autoimmune to help group
Flare,
3?
non-flare. Yeah, you can help group 3.
First of all, to diagnose the autoimmune disease, they didn't look only the laboratory result. They also look with the clinical symptom. And about to distinguish flare and non-flare, they use the criteria called SLE score. Hello, this and anyone hear me?
Yes.
Yeah.
Okay. And in a SLE score, they have a list about both of clinical manifestations and the laboratory result. For the laboratory result, they including the low complements or the thrombocytopenia or the leukopenia. That is might be used uh, used for in this case to see that hello,
Yeah.
you hear me?
Yep.
Okay.
Yeah, so that's all right now. So group 3, from Tamon 1 answer. Can you identify from SLE score 24 parameter? There are some lab finding in here, and you can say the tentative is active or inactive SLE, flare or non-flare. There are the lab tests in here that you can add in the score, not just antibody in A or ANA. Which one?
Yes, sir.
Which one?
Yes, sir.
ESR, yes. What else? ESR is high or low in this case?
High.
High. What else that the doctor want to see? And they request the lab test. ESR. What else? Only ESR, and the patient pay just 60 Thai baht. What else? What else? Or any lab that the doctor requests and then can say pathogenesis, that is SLE flare or active SLE or active autoimmune disease. Forget SLE, autoimmune disease, active autoimmune disease. Any lab that the doctor can see from your MT lab, send to them, and then they say, oh, this patient in the active autoimmune disease. Any lab, ESR, okay, inflamed. What else?
ANA,
ANA, you said antibody very low. You said, okay, maybe tentative. Just be careful underlying. What else? Flare, that means have the symptom, active.
Excuse me, I will help group 3.
Yeah.
One of most found clinical manifestation is about the nephritis.
Nephritis, but you don't see the patient from lab finding. Which one is tell you about kidney function? MT, please.
BUN creatinine.
Okay, what happened? BUN creatinine.
In this patient are normal.
What does that mean? No pathogenesis at the
At
kidney.
the kidney, yes.
Okay, so maybe it's not lupus nephritis, right? No complication at the kidney. What else?
LE cell in blood circulation.
I don't
Blood
give you
plasma.
LE cell from lab finding that your doctor request. You want like to the patient pay more money? No, I give you like just to package this. What else? You are searching, right?
Leukocypemia or not?
Leukocypemia. Why? Fair patient is have the leukocytopenia?
Lactate dehydrogenase because they reflect the tissue damage.
Tissue damage maybe it's not active SLE, right? In general, right? Tissue damage, yeah. Or we leave this question for the assignment 2, Ajahn Oh, because we are running out of time.
Maybe better.
Okay. Ajahn Oh, can you help me to contribute? I will list the question.
So yeah, so the thing that you missed is autoimmune disease differentiation or monitoring in these cases, right? And I'm not sure if the class has question or not. Maybe we can have questions from the class. Right? Any questions? Yes.
You correct. Our body try to regulate. But then the question is why L10 is just normal? Do L10 should be high? Always? Or high in what case? My COVID-19 or severe COVID-19? Do you have the evidence? The higher level of L10? If high is good for the patient, yeah, I agree with you. If high, I think it's good for regulation, right? But then think another way, maybe high is not good for fighting the virus because it's anti CMAR function, something like that, right? So it depends on the status of the patients. But in this case, IL-10 is not high. it's just normal. So what happened in the patient that we don't know?
I I I think IL-10 is normal because it's just 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 infect the COVID-19 is not to the state of the compensation. Yes,
It's possible.
possible.
It's possible because this is a real one that happened in the patient, but it's
Yeah.
just so normal. Yeah, even IL-1 beta is normal, right? But from your the first group presentation, 1 beta is pretty high, right? From cytokine storm, from the monocyte function, right?
Right.
But then
Yes.
the lab is not always high, low, right? When you work in a hospital, right? It's come from like this, right? Because it's even on the host status, right? Yeah.
Yeah, but I would like to add because from my finding, I have found that only in the severe case will have a high level of those the interleukin 10 and also the interleukin 1 beta because there is some some study has compared the interleukin level between the my COVID infection patients to the severe COVID infection, and they have found out that in the my infection patient have a significantly lower all of the interleukin level than the severe patients.
Yeah.
Thank you. Thank you.
Yeah. So I think we discussed a lot about it and try to link to the immunopathogenesis and also the lab finding. Sometimes it's not directly or clearly how the level of the cytokine in one patient will affect to the clinical symptom. So I think this is because of the dynamic of the immune response as well, which related to the clinical symptom and also the biomarker that we found in the in the patient. So I think so far now I'm not so sure that we really have the lab data that indicate this is the COVID infection apart from the molecular technique, right? Yeah, and also the clinical symptom. So I think if we would like to add up on this, maybe you guys can search for the data or the meta-analysis of all the publication out there that what are the biomarker that can tell this is the COVID infection, and even more appreciate if you can find that what are the biomarker of mild and severe symptom of the COVID patient, right? Yeah. So I think this thing that would be add up later.
Yeah. Yeah, because they have 2 weeks to find out more. อาจารย์โออาจารย์ชนิยาอาจารย์โอนฤทัยอาจารย์นี้ 2 weeks they need to give us more information. Today it's just overview what happened. So can I say that from the lab finding this is my COVID-19? Can I say that or severe COVID-19?
It's a mild case.
My
Yes.
non-severe, yeah, non-severe and with underlying autoimmune condition. All right, you guys hear this? Yeah, you agree, right? You agree with me? Okay. If you agree, then you need to work on assignment 2.
Because we didn't find, correlate with other clinical condition. We just roll out the SLE and then we continued with COVID. So we have to find other condition.
Sure, sure. So that's why อาจารย์ชนิยาอาจารย์โอ our staff we gonna finish for the assignment 1 because it take more than 3 hours. Students are so tired and me too. So then we have the assignment 2. That is more having fun. I'm so excited. But then our staff have the comment or discussion before we are talking about the next assignment.
So one thing that I think we miss for the group 1 too, because when we talk about a cytokine storm, so every group mentioned about it and explain a lot about the mechanism of it. But one mechanism that very important to induce cytokine storm from T cell is the super antigen, right? So so far we don't have any information about the super antigen in COVID or SARS-CoV-2 infection. So maybe this could add up for the next assignment. What do you think the super antigen will play a role in the COVID-19 disease?
Yeah. Dr. Poom, do you remember that from your presentation you talk about the he's there? Okay. You talk about the hyperactivation of T cell, something in the slide. So you find out more that the keywords is come from super antigen or not of SARS-CoV-2. Hyperactivation of T cell or lymphocyte, something from your presentation. You should go more in the detail that because of super antigen or not. You get my point?
Yeah, just find more.
Yeah. Yeah, Dr. Poom. Yeah, you get.
I just missed it and the signal just lost. So what happened?
Sorry. From your
sorry.
presentation, one slide that you show us, I think from the nature, have the word of hyperactivation.
Yeah.
Yeah. Please go out and find out more that the keyword is come from super antigen of SARS-CoV-2 or not. Because the word, hyperactivation. And from the picture, I see different color of lymphocytes. So I guess maybe it's come from super antigen, but I'm not sure. I don't have evidence. That is your homework. And you share with us for the next 2 weeks. Actually, yeah. What else that we should give the question to students to find out and come to class to discuss for the next time? First, super antigen. Yeah, sorry.
Yeah, so just to add up for more basic knowledge about the immunopathogenesis super antigen, that would be one thing. The other thing is because we touched upon about on autoimmune disease, especially in the systemic autoimmune disease SLE a little bit, but still we not so clear about the lab finding and also the criteria. So maybe next time you can present about how to diagnose the SLE patient in a full story, right? And then another thing is to move a little bit on the COVID-19 patient. As I mentioned earlier, that we don't, so far we still don't know that what is the biomarker that can clearly identify this COVID-19 patient apart from the nasopharyngeal swab. So maybe you can try to find if there are meta-analysis out there that try to link all the biomarker together with the COVID-19 patient. When I say biomarker, it's not just only cytokine, no? It's everything that we can find on the lab order. No?
Okay, อาจารย์โอ
Okay, I think
male and female and then อาจารย์นี้
okay, to me, I think the thing that you have in the slides, I think maybe like or you have everything in the slides, but you need to understand it because sometimes you just read it, but I'm not sure you can understand it, right? And to me, I'm quite impressed the way Natapumi present because he has no background in MT, right? But he can explain what he has in his mind, like what he understand, right? But many of you just read from the slide, but when we ask questions, we can see that maybe you just read it but you don't understand it. So the content, the content is important, but the understanding is the most important.
I do agree. So because if your basic knowledge is limited and then you can't go deep much, but then anyhow you can get the idea, right? Yeah, so I think Natapun can do Machima also quite good. Nashat, you excellent already on your knowledge. Just yeah, try to practice more. And Chimila also really give not too very much information. Yeah, but just looking back, how to say, look at the perspective view a little bit. So then I think that would be perfect. No, yeah. I cannot mention everyone, but this is just to pick up the example that how the thinking process and how the discussion could be continue in our class. Mm. No. And how about you, student? Do you want to say something or
more comment from
I'm not very clear
Ajahn
about
O?
instructions.
Chimila? You
Uh,
are about to say what?
I'm not very much clear about the instructions this time, and I have difficulty in understanding what I have to do actually. It happens to me. I don't know about others. And at the last moment also, I was not able to understand the question well and how I have to do it. I have that difficulty because I was reading research paper, I was reading question, and I was not getting the things which I need to.
Mm, I can't hear you well, Chimila. Yeah. I don't know everyone can hear or not. Maybe you can discuss and respond, but I can't hear you well. Mm.
What I was
I just come back.
I think she said she couldn't understand the assignment, right? Is that correct, Chimila?
Yeah, I was not able to understand what I have to do clearly. And since lab finding correlation with clinical manifestation, there was no clinical manifestation given on the paper in the lab finding now. So at the last moment, I was suffering whether the lab finding I have to use from the paper or lab finding given in the lesson plan. In our group, we had that problem.
Yeah, but actually,
Yep.
yeah,
Ajahn O, go ahead.
so like actually when we discuss it's quite late, right? Because last night we have a discussion, we had a discussion, but it's quite late. But the way that we plan this activity, we want you guys to carry the content from question 1, question 2, and then add up with the lab finding in the question 3, right? So that's what we want. Like we don't want to separate the story, but we want to make it in the sequence in this order, right? From question 1, 2, and 3. So you uh, have to think about what the group 1 gonna say, right? They're gonna talk about the clinical manifestation and what happened in the patients. And then in the question 3, you have to show the evidence that yes, this is actually happened in the patient. So that's the thing. But maybe we had discussion too late, so we don't have time to go over it much.
Yeah. The reason that why I don't give you the clinical manifestation because I think that you work in the lab like empty. You don't meet the patients. But then from the knowledge that you have from the first and second group, you can explain a little bit what happened in the patients. You can say possible maybe that happened in the patients, the abnormality like coagulation, the abnormality in the kidney or in the liver, something like that. From the lab, go to pathogenesis. Not always come from the clinical manifestation and go to pathogenesis. You know what I would like to say? Yeah, from the lab, you see the lab, you work like an empty. What happened? You can say what happened for pathogenesis in the patients from the lab. So the doctor gonna integrate that what happened because they meet the patient. So please understand my objective that I would like you to look at the lab and say what happened in the patients by use your knowledge from the first and second group. All right. Yeah. So then I need to finish and then to make it clear for the assignment. After this, you need to send the report to us because Ajahn Khao, I don't know full name. She would like me to have the score for the report. Can you summarize from your presentation and your friend presentation in one page? So you come with the writing or draw the picture or any mind map, whatever you know. You are grad student. The first group, second group, and the last one bring the knowledge and then send us the report. Only one page. So that means I don't want to read how L6 mechanism. I don't want to read in the detail. I'm so lazy for reading. But I want to see the knowledge that what can you integrate from the pathogenesis in COVID-19, in autoimmune disease, and lab finding. Let's say, if you think inflammation, cytokine storm, so then what happened? Clinical manifestation, what happened in this patient? And then what is the lab finding to find that pathogenesis, something like that. So you integrate that knowledge in one page, not more than one page. If more than one page, I don't read it. One page with single space, normal layout. Okay, we are lazy to read your report, so then you need to synthesize, integrate the knowledge, and then summarize for us. One page only from 3 topics: immunopathogenesis in COVID-19, lab finding, and link to autoimmune disease. Okay? Yep.
May
Send
I
us
ask about the,
to what is the question?
The font size and which font?
Front side? Okay, front side. I'm too old. So then, ask the senior, Ajahn Chaniya. What size do you need? Front side, Ajahn Chaniya.
Because you're gonna send us as a English one, so then Tamil Roman 12 would be fine.
Okay, Tamil Roman will be fine. Send us before 14 October. All right. Then we have assignment 2, right? From the lesson plan. Assignment 2. We have 2 questions that Ajahn O give you about the lab finding. From the lab finding, group 1. How many students do you have in group 1? 1, 2, 3, 4, 5, 6, 7. You have 7 people in group 1 to answer. Yeah. The first one from lab finding. Please explain us more about pathogenesis from lymphocytopenia and anemia. Don't come with the words like lymphocytopenia because T cell apoptosis. No. How T cell induce apoptosis? How virus induce T cell apoptosis? Okay. Go to in detail. Why the patients have anemia? Don't tell us like ferritin is decreased. No. Tell us about pathophysiology plus pathogenesis. All right. Group 2. Biomarker for inflammation. CRP, ESR, procalcitonin, and inflammatory cytokines. Integrate together in the package of inflammation and then explain the pathogenesis. Don't come like CRP is inflammation from the liver. No. CRP, ESR, procalcitonin, cytokines, it come together and explain what happened in the patient. You can have 1 slide, 2 slides, not many slides, okay? If you are good as the MT. This is your assignment 2. You come and have a class discussion on 21st October, right? Yes. And then from the question that Ajahn give you, Ajahn O and Ajahn Chaniya, I mean the staff, super antigen is go to group 1. Immunopathogenesis in SLE and the scoring for saying active, non-active go to group 2. Biomarker in COVID-19, both group. Yeah. Biomarker in COVID-19 in both group. You share the knowledge. If your friend in group 1 find biomarker A, then you present biomarker B. All right. You need to talk, work like a teamwork. Yeah. All right. And any questions?
อาจารย์คะ about the pathogenic of SLE อาจารย์ mean how it's correlate to the laboratory results or the criteria, right?
Yes, madam. Huh? What is the question?
Can you type, because for the writing we may difficult to read some type of writing. Typing is quite easy for us to check your spelling or check your concept.
Yeah. You can draw the picture for chart, my map. The score is up to your attention. Okay, more question? Similar, you understand my assignment, right?
Yes.
Okay.
อาจารย์คะ if we have something like the reference, could we add the page to add the reference?
In the report, right?
Yes.
If you add the reference, you add in the second page, not in the first page. Or you want to reference like from your writing? Or you add reference after? Which one you prefer?
I prefer reference after.
If after, you can put in the second page.
I doubt about the reference because this is to summarize what we are talking today because we want to get a score from you. Maybe you can just type it down what you understand from today. And I don't think that you need
reference, right?
Yeah.
Nah, you need reference from your friend presentation. What do you mean? Or you like to read more and then you make up your idea more? You have time for that?
It's only one page. Would be like yeah.
The group share the slide.
Summarize, yeah. You know summarize.
Can the group share the slides?
Well, it is
summarize just only
summarize from our understanding.
Yes, summarize just only key finding from your knowledge. That's all. You can send us like 10 sentences. That's fine. Key finding. Yeah. More questions from students before we leave the class because we are so tired.
Can the SLE and other group slides connect to the Google group?
Three groups.
Can the slides from other groups can be added to the group?
Yeah, you can
share.
Sure.
I think you share already, right? Or you don't see the slide from your friend before come to the class?
In the chat, in just only their group, not between the group.
Oh, please talk. Please talk.
So Shamila want the slide from other group, right?
Student, please share your slide and your knowledge before come to the class. It's not confidential. It's not the top secret. You share slide with your friends, share knowledge before come to the class. Okay? And the slide today, you should share with your friends. All right? Even article, knowledge, any material that you have, please share with your friends. More question?
I think on the lesson plan, I'm not sure that the uh, name in group 2, maybe we meet someone, so I'm not sure about that.
Oh, really? Sorry. Who I miss? Who I
กัน
miss?
ทอน
Oh,
Uh,
sorry.
อ่า
there are two Tamon one.
Yeah, Kanton and who else?
Me too.
Now part who
and maybe เศรษฐวุฒิ
It did have two.
Oh, อาจารย์ Oh, this is the last version that you get from the Google Classroom, right?
I think so, but I'll check it again. But
No. This
No.
version is not in the Google Classroom.
Already post the new list.
Yeah.
Oh, sorry.
Yeah, but Ajahn O can do this. Thank you. Yeah. So to make sure that student get a good teamwork.
So the first group, you have to put the Shemila in the first group. First, yep. First group is Mongkol Natapul Benjamat Chayisara Nashat Pipitak Shemila.
Okay.
And the second one, Tamon one, Nishakan Nashima Jirachaya, right?
Jirachaya.
Yeah, Jirachaya, Kanthor, Napatipu, and Setut.
Yeah, that according to the Google Classroom that I update. Yeah. All right. Can we just finish and get done with this? Sorry, class too late. Almost 4 hours. So if no question, we're gonna say goodbye and then see you next 2 weeks. Yeah. Thank you so much for give us the knowledge. Actually, we are learning from you. So it is a new topic. The one thing that I would like to make is clear that when you study in the grad student, sometimes it's just sharing the knowledge. You could not say that is a correct, this is incorrect. You just say, how can you explain that? So it's come in the discussion. Sometimes we don't know, and then you know it, just share this discussion. All right? Right. No need present hypomaturation in the SARS-CoV-2. I don't know before. I learning from her, something like that. So class discussion is very important. Don't be afraid to ask the question and share with us. For me, I don't know many information that you share with us, so it's okay. I think if I don't know, I say I don't know, and I want to know. So please talk with us, share with us. This is the way that you can learn in the class. Yeah. อาจารย์โออาจารย์ชนิยา you have more? Then no.
No, but I agree with
อาจารย์ชนิยา you want to say goodbye or you want to say something?
I want to say something. I do agree with Ajahn Pashani on the point that
All done, right, Ajahn?
Oh, I'm talking. No, just keep in mind that we are grad students, so then there will always new knowledge that come into the field. So just keep searching and reading and then discussing.
Yeah. Okay. Bye bye. See you next 2 weeks. Yep. Thank you so much for today. Yep. Bye, Shimila.
Thank you ค่ะอาจารย์
Shimila, are you the same time zone with us?
I didn't get your name.
Are you the same time zone with us?
No,
No. Are you ahead or behind?
behind.
Behind. So it means how many hours?
For me, it is now 4:50.
4:50. It's the same. Me also 4:50.
2:50. Sorry, 2:50. Now I
5:50. So it's 1 hour.
2:50.
2:50. So we are ahead of you for 4 hours. No, 2 hours. Okay. So then you
Okay.
have more time to talk. Okay.
Bye, guys. See you. Thank you so much.
Thank you.